Hi everyone, good evening guys. Welcome back to the channel. This is Unacademy NEET English channel. My name is Tina and I'm your biology educator. Good evening all of you. Good evening. So guys, this is part 2 of PYQ sessions. We are discussing lots of questions on YouTube and also on special classes. So practice is must, and guys, there are just 2 more days left, that is Thursday and Friday, for your mock test series. Do not miss these 2 tests. They are going to play a very crucial role. All right. So please let me know that you're all set for the session and we can start with the PYQ discussion for class 12 today. Good evening. Good evening. So guys, I am going to start from the last chapter, that is ecology. I'm going to start with the questions of ecology. Quickly, let me get a thumbs up that you're all set for these questions. Yes, please. Let us start with this. All right. So we're going to go backward to forward. All right. Let's begin with the session. Okay. So very important. Just a second. Please set few things. All right. So it says habitat loss and fragmentation, overexploitation, and alien species invasion and co-extinction are the causes of. So what does all these reasons do? They lead to biodiversity loss. They lead to biodiversity loss. The answer is this. And you will see, I'm discussing last 5 years questions. You will see that this habitat loss thing is coming again and again. Is that clear? Now, in situ conservation, second most important thing from this chapter is in situ conservation. In situ conservation refers to conservation and protection the whole ecosystem. So basically, in situ means organism is going to be protected within its natural habitat. So when you are protecting the natural habitat, you are protecting the whole ecosystem around the organism. Is this clear? Right. Moving on to next, which of the following is not a method of ex situ? See, you're getting questions on in situ and ex situ since 2022. Every year you're getting some or the other question. Not an ex situ. So not an ex situ is national park. Rest all are ex situ. That means organism will be taken out of the natural habitat and protected elsewhere. Be it micropropagation, which is known as tissue culture, cryopreservation, where we have minus 196 degrees Celsius temperature at liquid nitrogen, seed banks, pollen banks, sperm banks, all these represent cryopreservation. And then in vitro fertilization. These are all ex situ, while national park is a natural habitat. So it comes under in situ. Are these points clear? Now, which of the following are true about the taxonomical aid key? Now, see, keys are not in your syllabus, so you can just skip this question. Next, Western Ghats have a large number of plants and animal species that are not found anywhere else. What term will you use to notify such organisms? So, such organisms, guys, are known as endemic species. Endemic species means which are restricted to a specific area. They are not found anywhere else. They are not found anywhere else. Is this clear? Okay. Moving on to frugivorous birds are found in the large number of tropical forests, mainly because of. Why are they found in the tropical forest? Because of the availability of fruits around throughout the year. So you need to have that food for the given species. So if frugivorous is present in a particular area, it means that food is available. Next, moving on to this question, again related to in situ, ex situ also. Sacred groves, guys, sacred groves. Example of sacred groves is Khasi Hills. You have to learn the examples of these sacred groves also. Zoological parks. Zoological parks are, they represent the ex situ conservation. Nile perch. Nile perch resulted in alien species invasion. And Amazon forest, because they occupy large area, they release large quantity of oxygen. So answer will be B. Are these points clear? All right. Moving on. Carbon dissolved in the oceans. What is the amount of carbon dioxide dissolved in the oceans? That is 71% carbon dissolved. Annual fixation of carbon during photosynthesis. That is around that is around 4 into 10 raised to power 3 kgs. Then par is just 2 to 10% of the total insulation. And then productivity of oceans is 55 billion tons. Now you may not be sure about this value, annual fixation. Then use the elimination method, use rest of them and just connect the leftover value with annual fixation. Is this point clear? All right, moving on. Now, World Summit. There are just two summits here given. One is Earth Summit and second is the World Summit. World Summit was on sustainable development in 2002 in Johannesburg in South Africa. So the question itself has many things that can become question for the next year, right? So what did they pledge for? They pledged for significant reduction in the current rate of biodiversity loss at national as well as local levels. At every level, they have to cut down the reduction in the rate of biodiversity. Is this clear? Not going to continue revision classes, Mahima. We are left with very less time, so that is why I knew, and I have completed NCERT reading already. So that was my priority. That was my priority. I would rather suggest, guys. So you have Friday, Saturday, sorry, Thursday and Friday. I won't suggest this for Saturday. You have tomorrow and day after tomorrow. I have completed NCERT books in just 4.5 hours. You can increase the speed and complete your NCERT reading for the final time in 2.5 to 3 hours. So just go through your whole NCERT and make sure. Now just see these PYQs, uh, solve these PYQs, and now when you go back to NCERT, the things will click in a better way. Is this clear? So make sure that you do that once more before going for the exam. Again, coming on to the causes of biodiversity loss. See, causes of biodiversity loss is another repetitive topic for in your NEET exam. So what are the causes? Overexploitation, habitat loss, and fragmentation. Now, whenever they're using the word only, you should be overcautious. Usually, only options, the options with only and always type of words are not the answers. Now, species of plants. Also, guys, I have just put a video for you in case you are unsure about the question, how to guess. How to guess. Now, see, guesswork is slightly different in biology. I would suggest, do not use the same strategy which you use for your physics and chemistry. So go through the video. I've specially made it for biology because I know there are so many videos out there, but every technique doesn't work in biology. So what things you have to take care while, putting your guesswork, then please be cautious. Okay. Yeah. Now, the species of the plants that play a vital role in controlling the relative abundance of other species. What do you call that? Keystone species. What do you call that? Keystone species. Now, alien species which we know, they don't belong to the particular area, they are not native, they've come from the outside place. And then another characteristic of alien species is that it is going to eliminate the native one. Endemic means it is restricted to only a specific area in the whole biosphere. Exotic again means which has come from outside, but exotic does not necessarily mean that it will eliminate the native species. But alien species is exotic, it doesn't belong to the native land, and it is going to put the native species in danger. Are you understanding the different terms? Hi everyone. Moving on. In rivet popper hypothesis. Now, rivet popper hypothesis, who gave it? Paul Ehrlich. Compare the rivets in the airplane to. Very important. What are the rivets being compared to here? The species within the genus. And okay, another question to you is, what is in rivet popper hypothesis, which analogy has been used to compare with ecosystem? Answer. Yes, please. Which analogy has been used by the rivet popper hypothesis, which has been compared to ecosystem? Let me know if you know the answer. If you know this answer, seats or the whole aeroplane plane? Yes, the whole aeroplane has been compared to the ecosystem. That can be another question for 2026. Yes or no? So, historic convention of biological summit, that is Earth Summit. When was that held, and that too in Rio de Janeiro? Answer is 1992. So guys, first happened Earth Summit. It was followed by World Summit. So on both the summits, the question has been asked. So you should be ready with that. So any question can come up again. Now the evil quartet, see, causes of biodiversity is one hot topic for NTA. They are asking one or the other thing about evil, about the causes. So evil quartet which one is the most important cause of extinction? The most important cause is always the habitat loss and fragmentation. You will see this question coming up again and again. Now, list biodiversity hotspot, sacred groves, then Johannesburg, and then alien species. Now, alien species is Parthenium. One alien species is Parthenium. Johannesburg, South Africa. This was the World Summit. So we have got the question in 2022 also. This is in 2024. Now 2022 also had the question on this topic, right? And then sacred groves. Where do you find them? Khasi and Jaintia Hills in Meghalaya and other places. So make sure. Now, guys, do one thing. Keep a pen and the paper along. Keep noting down the questions, the topics that you see recurring again and again. That is very important. This is going to be very crucial now. What are you going to revise for the next 2 days? These are the things. Okay, biodiversity hotspot. Hotspot means the area has species richness and some endemic species. That area is known as a hotspot. Western Ghats are one of those. Then moving on to cryopreservation. Already got a topic question on cryopreservation, right? In a different format, cryopreservation is used for. It is used for the preservation of gametes in the viable and fertile conditions for a long period of time. Let me know in the chat, guys, what type of conservation does cryopreservation present? Is it in situ or ex situ? Let me know quickly. Hi, Akina. Is it ex situ or in situ? Is it in situ or ex situ? Yes, guys. Stargirl, it's a time for complete NCERT and I've already done that. So please focus on this and solve more and more questions, guys. I have already taken 10 question round classes on special classes. We've discussed so many questions. XC2, very good. It represents the XC2. Moving ahead, which of the following is not included in C2 once again? Which is not included? Earlier, they were in 2022, they were asking which of the following is not included in XC2. Remember, now they are asking they are not included in C2. So, wildlife sanctuary, botanical, biosphere, and national park. What is the answer, guys? Botanical gardens. Why? Because botanical garden is a XC2. You take the plant from the natural habitat, grow it somewhere else, and then you conserve it. That is botanical garden. Now, highest annual net primary productivity is observed in net primary productivity (NPP) is observed in the biggest area possible, that is tropical rainforest. They represent the highest productivity. Then regions with high level of species richness, high degree of endemism, and a loss of 70% of the species and habitat, they are identified as. Just discussed it with you that a hotspot is an area which has maximum species richness and also some endemic species, high degree of endemism also. And sadly, they are facing a threat of extinction. So loss of 70% of the species has been noticed from these hotspots. Answer will be biodiversity hotspots. Can you answer me in the chat how many hotspots have been declared declared so far? How many? How many? Yes, how many hotspots have been declared so far? Let me know in the chat quickly. Now, identify the place, guys. I am very fine, Akila. Alikya. Okay, please concentrate. Identify the places from where the sacred grooves are not found. So, sacred grooves is also a topic which is recurring again and again. So, I'm sure that you are taking down these points. 34. Yes. How many hotspots have been identified in the world? 34 now. So, sacred grooves I'm talking about, they are also very important question of the chapter. Aravali Hills, Chanda and Bastar area of MP, Mudumulai Sanctuary in Tamil Nadu, not found, they are saying, and Khasi and Jaintia. So, this is true. This is a sacred grove. This is a sacred grove. This is a sacred grove. C is not a sacred grove. So, answer would be C only. Is that clear? Alright, given below are the two statements, guys. Rainforests which cover more than 14% of the Earth's land area, now reduced to 6%, and Amazon rainforests have greatest biodiversity on the earth. So here we see that both the statements are absolutely true. Answer will be A. Answer will be A. Are these points clear? Alright, moving ahead. Now guys, please see the trend. So far we have been discussing the questions from ecology and that to the last chapter, biodiversity. Over time, guys, ecology, genetics, biotechnology, they have gained so much popularity amongst in NTA world. So last year we got in total around 10 questions. Think of it, 10 questions from ecology. 10 means 40 marks from ecology itself, right? So very important to focus on this. But the trick is that these questions are recurring, coming again and again, right? So there are some typical topics. So take notice of that. Okay, Robert May. Robert May said that global species diversity is around 7 million. Alexander Humboldt gave us a species area richness, sorry, relationship when he gave this type of graph, right? And then Paul Ellery gave us rivet popper hypothesis, and David Tillman performed the long-term ecosystem experiment using the outdoor plots. Are these points clear? Are these points clear? Okay, I'll be putting this scientist video very soon for your last minute revision. That will be a quick video. That will not very detailed. You can quickly revise all the scientists. So I'll be releasing it very soon. Now, type of conservation in which the threatened species are taken out from their natural habitat. You take it out from the natural habitat, put it in a special setting where they can be protected. What do you call it as? What do you call it as? Yes, it is known as, when it is taken out from the natural habitat, this is known as XC2. This is known as XC2. Now it is not given in here. So it is not in C2, it is not semi-conservative, it is not sustainable. You will just write it biodiversity conservation. Is that clear? So answer should have been XC2 to be very precise. But now when it is not given, you will go with the B option. Now, 2024, the major causes of biodiversity. Do you see that every year NTA is giving you question from major causes of biodiversity? So this question is pakka pakka in your paper, right? Now, overexploitation, co-extinction, mutation, habitat loss, and migration. So you have to tell me the causes. Causes. Overexploitation, yes. Co-extinction, yes. Habitat loss is a major reason in the evil quartet. Not mutation, not migration. So answer would be D. Are these points clear to all? Now, tropical regions show greatest level of species richness because why have there been highest level of species richness in the tropical area? Now there are 3-4 points given in NCERT. Let's see. Because they have remained relatively undisturbed and hence more time for species diversification, right? Second, their tropical regions are more seasonal. No, they are not seasonal. They have a constant season. Then more solar energy is available. So this is not the answer. More solar energy is available in the tropics. More solar energy means more productivity. More productivity means it can support higher level of or higher number of species. Are these points clear? Answer. And then constant environment promotes niche specialization. Clear cut NCERT line. And they have been constant and predictable. Unlike temperate regions, which have suffered lots of fluctuations in the environment, glaciation, right? And they receive lesser amount of solar energy. So answer would be A. Are these points clear to everyone? How frequent are the numbers data asked from biodiversity? Not very much. You have just seen, not very often. So that should be the last priority for revision. Now, when you're doing the selective revision, these are the points that you have to take care of. Okay, and I would suggest strongly that please give NCERT a quick reading again. Spare out just 5 hours, and in 5 hours you can complete 11th and 12th NCERT reading. At least the most important lines from where questions are coming back again and again. Is that clear? Yes. Spare out 5 hours for biology, quick revision, whole NCERT. Now, list of endangered species was released by who's responsible for all this? It is IUCN. It is IUCN. What does GEAC do? It is related to checking the validity of the research of biotechnology, right? WWF, we don't care. Foam, we don't care. Not in our syllabus, right? Answer will be IUCN. Now, evil quartet. What does evil quartet do? Evil quartet are the quartet means 4. So these are the 4 reasons which or causes of biodiversity loss. XC2, where it is taken out from the natural habitat, it is cryopreservation, right? And then we have lantana. Lantana is an alien species invasion. In another year, I think in 24 or 23, they have given parthenium. So this year, in 25, last year, sorry, they have given this example of parthenium, sorry, lantana. So Parthenium, lantana, these are the alien species. What is the third example given in NCERT? Quickly let me know. What is the third example given in NCERT? Yes, guys. Who remembers the third example of alien species invasion? Yes, Parthenium is done already by 2022 or 23. 25 has asked about lantana. Now, 9, that is not, yes, it is alien species invasion there, but I'm talking about plants. In case of plants, guys, it is Ishornia. Very good, Asha. It is Ishornia. So, if you are able to think about all these questions that I'm quickly asking, so you are on the right track. Just keep a calm mind and go for your exam. And then dodo has already been extinct. Now, which of the following is an example of XC2C? Causes of biodiversity and methods of conservation. These are two very important things. These are two very important things. Are you getting these points? Please let me know. So guys, even I am taking notes of all these points. In fact, I've already taken and I'm going to share it with you also at the last moment. But please make sure that you are writing down these points yourself. Is that clear? You are writing down these points yourself. So, yes, please. So one is causes of biodiversity loss, yes or no? And second is methods of conservation. So these are very important points from these chapters. Are you clear? Are you clear? Moving on to the next. So it says, what is an example of XC2 conservation? Which is a method of XC2 conservation? Yes, please. Okay. What is an example of XC2? It is zoos and botanical gardens. Rest all are in situ. Rest all are in situ. Alright, moving on. Which of the following is not correct? Not correct. Pyramid of energy is always upright. Now, pyramid of numbers in the grassland system is, in the grassland system, ecosystem, numbers is upright. Then it says pyramid of biomass in sea is generally inverted and pyramid of biomass in sea is generally upright. See, we know energy always goes on decreasing from lower to upper trophic level. So this is 100% true. Energy, for energy pyramid, we don't have any other option. It is always upright. So maximum at the producer level and goes on decreasing as you go upwards. If we talk about the number in the grassland ecosystem, so just think of a lot of grass. Grass here represents the producer. Then there will be some herbivores and lesser number of primary carnivores, even lesser number of top carnivores. Yes or no? So the number goes on decreasing in the grassland, and that is how you get a upright pyramid. Then if we talk about biomass in the sea, see, it always starts with the producer level. So producer in seas are planktons. They are planktons. That could be algae, right? Now see here. So planktons, then planktons will be eaten by small, we have, sorry, zooplanktons. Zooplanktons are eaten by small fishes and then eaten by large fishes. Are these clear? And then pyramid of biomass in sea is always upright. So we are talking about the biomass here. So no matter if the large, see, now listen to the question. Now this is the pyramid of number. This is the pyramid of number in the sea. If we see the biomass, it has to be inverted. No matter how many planktons are there, but their body mass, their biomass will be lesser. Even if there are millions, then the biomass will be lesser as compared to the zooplanktons or the small fishes. And then one big fish is enough to compete with the biomass of so many small fishes, right? So this is how you get the inverted pyramid of biomass in sea. And then it says biomass in sea is generally upright. This is a wrong statement. Moving on to the next question here. Moving on to the next question here. Just a second. Yes, please. Moving on to the next question. Now, in the equation GPP minus R is equal to NPP, what is the R? What does R represent? Very easy. Respiratory losses. Now, the amount of nutrients such as carbon, nitrogen, phosphorus, calcium present in the soil, guys, at any given time, this is known as, see, these are all inorganic substances. These are all non-living substances, right? Non-living factors. So this is known as standing crop. Has it been about the living component, it would have been non-living, it is standing state. For the living components, the answer would have been standing crop. Are these points clear? All right. Now, detrivores break down the detritus into small particles. What is detritus? Anything that will undergo decomposition. That is, raw material for decomposition is known as detritus. Now, detrivores are going to break it down into smaller pieces, large pieces to small pieces. This is known as fragmentation. This is known as fragmentation. What about catabolism, guys? Complex molecules into smaller molecules, right? Okay. Now, amount of biomass or organic matter produced per unit area over given time by plants. So when we are talking about plants, that is the producers, then, okay, this is the day my pentab has chosen to disobey again. Okay, the amount of biomass or organic matter produced per unit area over the time by plants. So about producers, it is always primary production. It is always primary production. Are these points clear? Please let me know that these points are clear to you all. Is this point clear to you all? Yes, please. All right, moving on to the next. Again, see, now from this chapter, GPP, NPP, that is production and productivity, is a very favorite topic of NTA. Do you notice that every year you are getting question out of it? Now, GPP minus R is equal to NPP. This equation is just learnt by us by heart. So GPP is gross primary productivity, NPP is this, and once again it is asked this exact same question. What does R stand for? Respiratory loss. So moral of the story, you will find questions from the PYQs. So see, it is like revealing the paper. You know, PYQs is one tricky way in which, very interesting way in which NTA has already, yes, announced the paper. So you solve these questions religiously, and you are certainly going to see some questions coming back again, right? What better than this? Identify the correct statements. Detrivores perform fragmentation. Humus is further degraded by some microbes during mineralization. Then it says water soluble inorganic nutrients go down into the soil, get precipitated. This is called leaching. Then the detritus food chain begins with the living organisms. Is that so? Then earthworms break down detritus into small particles by a process called catabolism. So which of the statements are correct, guys? Detrivores are going to perform fragmentation. And we know that earthworm is a detrivore. So when it breaks it down into smaller pieces, it is known as fragmentation and not catabolism. It is called fragmentation and not catabolism. Is that clear? So this is wrong. Detritus chain does not start with the living organism. It starts with the detritus. It starts with the detritus. Are these points clear? So these are the two wrong statements. So answer would be A, B, and C. Moving on, which of the following is not a limitation of ecological pyramids? So this is the last paragraph of this chapter, where they have clearly mentioned 3 to 4 reasons which are limitations. That means what are the things which cannot be concluded from the ecological pyramid? See, the first limitation is saprophytes are not given any place in the ecological pyramid. Now we know the base will always represent the producer, and the apex will always represent the top carnivore, right? So saprophytes don't find any place in the ecological pyramid. This is true. This is true that this is one limitation of pyramids. It assumes a simple food chain and which almost never exists in nature. Again, true. Basically, in nature, what exists is a food web, but they don't consider food webs. It accommodates a food web? No, it does not. So this is the limitation. It does not take into account that same species belonging to one of the more trophic levels. Why? Because it is not considering the web part, food web part. Is this point clear? The answer would be C. Now, here you have been given the energy of the pyramid. So T4, it says, is equivalent to 1000 joules. What is the value of T1? So we know that there is 10% law operating in nature. So only 10% of energy from the lower trophic level is passed on to the upper level. So whatever energy T1 has, only 10% of it will be received by T2. And similarly, 10% of T2 will be given to T3 and 10% of T3 will be given to T4. So whatever T4 has is just 10% of what T3 had. So you'll just keep on increasing it by 10. That is the trick. So this will have 10,000, right? This will have 1 lakh, and then this one will have 10 lakh. Are you getting that? The trick is when you go upwards, you decrease 10, and if you go downwards, then you have to increase 10, and that will give you the value according to the 10% law. Are these points clear? Are you getting this? Have you gotten this trick? When to reduce 10? When you go upwards, and you increase increase the value by 10 when you go downwards. Is that clear? Okay. What is incorrect about an ecosystem? It can vary from small size pond to large size pond. Yes, it can be. It can be aquarium also. In aquarium also represents an ecosystem, or it can be represented by a large ocean. It can be anthropogenic. Yes, anthropogenic means it can be man-made. I just named a man-made ecosystem which happens to be crop or crop field or aquarium. It may be temporary or permanent. Absolutely. Now crop is temporary, aquarium is temporary, right? But it involves the function of flow of energy but not the recycling of nutrients. No, this is wrong. So guys, there are four characteristics, four properties of an ecosystem. Who can name all the properties? One of the properties is recycling. Then we have, yes, who can mention all the four properties? Properties of ecosystem. If you can't, then you need to go back to NCERT and revise it again. Make sure that you do that. Okay, moving on. The rate of decomposition is not related, it says it is not related to the chemical composition of detritus and climatic factors. In a particular climatic factor, decomposition rate is faster if detritus is rich in chitin and lignin. So basically, both the statements are false. Why? Because chemical composition is an important factor to determine the rate. How fast decomposition will occur depends upon the chemical composition. For example, if it is rich in lignin and chitin, then decomposition rate is slower. In statement number B, it has written the wrong thing. It is not faster, it goes slower. While if it consists of carbohydrates and proteins, it will undergo decomposition at a much faster rate. Is this clear? Now, so we have list tertiary consumer, secondary consumer, primary consumer, and primary producer. Now this can be one of the easiest questions that you can get. So we start with primary producer. So producer has to be an autotroph, which is grass. Then who eats the grass? Primary consumer has to be the herbivore. So answer would be goat. Then secondary consumer is one which eats the herbivore. It would be wolf, right? It would be wolf. And then tertiary would be lion. The top carnivore is known as top carnivore. Okay, top carnivore is the tertiary consumer. Again, NPP. See, are you seeing? Are you getting that? NPP. They're giving you this question again and again. And guys, now, since we have already read NCERT, we are already discussing these PYQs. Make sure that you solve the two papers left in AIMT by yourself. So spare these 3 hours tomorrow and day after, even if you have missed the previous test. If you miss these tests, I'm sorry to say, that can cost you very heavily. Before going for the NEET exam, you need to get into that zone. It is important, guys. See, I am very sure that you will understand this importance sooner or later, but the sooner you understand, the better it will be for you. Otherwise, regrets are the real things in life. Okay. Now, in an ecosystem, if NPP of first trophic level is 100x kilocalories per year, what would be GPP? What would be GPP? So, what would be the GPP, guys? What would be the GPP? See, that will be exactly equal to the GPP of the third trophic level. Do you understand how? See, if this is the first trophic level, this is the second one, and then this is the third one. So GPP of this is equal to the NPP of this. Yes or no? Right? So the GPP is the production, what is left over. So GPP of one is the NPP of other. That is why the value is exactly same. Which of the following is the unit of productivity? Now another very important question. Units of production and productivity. A very easy trick to remember. Production is not time based while productivity is time based. So in all the three there are no times considered, while only in the B option we are considering time. See, what is productivity? It is the rate of production. It is the rate of production. So whenever we are using the term rate, it considers the time factor. So it is only the B value which has time factor in it. Kilocalories per meter square per year. Is it clear? That is why answer is B and not D. Is this clear? Is that clear to all? And then it is per square, not per cubic meter. Is that clear? Okay, moving on to the next question. In ecosystem, guys, there is unidirectional flow of energy from sun, from producers to consumers. Then ecosystem are exempted from the second law of thermodynamics. This unidirectional thing is true, but second law of thermodynamics, this is false. So guys, our ecosystem will obey first law of thermodynamics, second law of thermodynamics, as well as third, right? So the first two have already been given in your NCERT. Who is known as the father of ecology? It's Ramdev Misra, the scientist uh, whose picture is given in the end of the book, right? The last unit. So it was Ramdev Misra. Now the primary source of energy in ecosystem is solar energy. Can you name the primary source of energy in underwater? Yes, underwater, which source of energy is a primary one? Yes, guys, primary one. Okay, the rate of you have to answer, guys. You have to no. This is for see. This is basically for terrestrial ecosystem. I'm talking about water. I'm talking about the yes. I'm talking about the aquatic ecosystem, deep sea. Are you sure? No, Asha. Mm-hmm. Think over it. Think over it. Now, the rate of production of organic matter photosynthesis in ecosystem is called the net primary productivity. Guys, this is wrong. So this is actually known as gross primary productivity. So produced by plants, organic matter produced by plants. That is by photosynthesis. This is what gross primary productivity is. Moving on now. So did you notice that you're getting questions from GPP and NPP again and again? Units of production and productivity is coming back again and again. Yes or no? All right. Okay, moving on now. In the exponential growth ecosystem, this is the value, sorry, this is the equation. What does E represent? Very easy. Very easy. E represents the base of natural logarithm. Base of natural logarithm. Now my question to you is, what does R represent? Yes, please. If anybody can tell this, what does R represent? If not now, then guys, make sure if you're watching it in the recorded form, you have to answer these questions in the comments. Is that clear? All right. Amensalism can be presented by, what is amensalism? One is harmed, other remains unaffected. So still, since one is harmed, it is a negative interaction. So it has to be minus and zero. Can you give me an example? Can you give me an example? Antibiotic, yes, carrying capacity. Okay. Antibiotic resistance. Sorry, not antibiotic resistance. Antibody production. Antibody production. So antibody will be produced by the organism who's meant to do that. Let's say penicillium produces antibiotic, but it is not affected by the antibiotic that it produces. Penicillium remains unaffected, but staphylococcus gets killed. So penicillium is unaffected, but so it shows 0, and negative is for the bacteria which gets killed. Next point. In spite of interspecific competition in nature, which mechanism the competing species might have evolved for their survival? Yes, please. Competing species might have evolved for their survival. So, see, according to Gauss, So there is, according to Gauss, one of the competing species will eventually get eliminated. But then we have another phenomena which says that nature does not favor elimination all the time. So there will be, and there has been an evolution of mechanism which will allow coexistence of the competing species. The, what do we call it? This is called resource partitioning. They will divide the resources and live together happily ever after. Now, 8 Drosophila in the lab population of 80 died during a week. So if 8 Drosophila in a lab population, so this is the original population, this is the original population, this is the number of deaths, this is the number of deaths, yes or no? The death rate would be, what is the formula for death rate? So number of deaths upon, excuse me for this bad handwriting, it is because of my pen tab. So number of deaths upon original population. So answer would be 8 upon 80, that is 1 upon 10, that is 0.1. Is that clear? Similarly, if there is a question, you will see the question in the earlier years, that would be related to birth rate. So birth rate will have the same formula, number of new births upon the original population. That will give you the birth rate. Is that clear? So death rate is deaths upon original. So everything has to be. So the common parameter around which we compare death rate or birth rate is the original population. Now, while explaining the interspecific interaction, plus sign is for beneficial interaction, negative is for detrimental, detrimental is harmful interaction, and 0 is for neutral. Which of the following interactions can be assigned plus or minus involved in the interaction? So that means one is benefited while the other one is harmed. So can you name can you name an interaction which shows plus minus type of interaction? See, we know evangelism, just read, it is, it is 0 and minus. It is minus 0. Commensalism is plus 0. One gets benefited, other is unaffected. Competition is a negative interaction, while predation is positive negative. So answer would be D. Answer would be D. Now, which of the following cannot be connected to predation? Cannot be connected to predation. Okay. It might lead to extinction of the species. Both the interacting species are negatively impacted. It is necessitated by nature to maintain the ecological balance, and then it helps in maintaining the species diversity in the community. So we've just seen that predation is a plus minus interaction. So the the predator, the stronger one, the predator is benefited while the prey which gets eaten is harmed, right? So predator and prey relationship are positive and negative kind of interaction. Yes, predation is plus minus interaction. So it says both the interacting are negatively impacted. This is true about competition, guys, not about competition. Sorry, not about predation. So this is the answer. Clear to all? Now, moving on to 2023 question. See, 250 snails in the pond. Within a year, the number increases to 2500. This is about birth rate. So what was the formula I just told you? Birth rate is equal to number of births upon the original. So earlier it was 250, then it became 2500. So what was the total number of organisms added? So it will be 2250. Yes. So divide 2250 upon 250. Can you find out the answer? It will be 25 ones are 25 fives are. Answer, sorry, 25 nines are. Yes, 25 nines are 225. Answer would be this. Is that clear? 25 fours are 100, 25 eights are 200, 25 9 is 225. So answer will be 9. Are you getting that? How to solve the questions? Now for chemical defense against herbivores, calotropis. See, organisms do not graze upon calotropis. They don't graze upon calotropis. Why? Because they understand that calotropis have cardiac glycosides, so it can lead to sudden cardiac arrest, and that message has already flown in the ecology, in the ecosystem. Sorry. Plants offer rewards to animals in the form of pollen and nectar, and animals facilitate the pollination process. This is an example of, what is it? Example of mutualism. Both the organisms are benefiting each other. Plants get pollinated, and as a return gift, animals get the food and nectar, that is pollen and nectar. So it is a plus plus type of interaction. It is a plus plus type of interaction. What do you call it? Mutualism. Mutualism. Is this point clear? Is this point clear to all? Now, given below are the two statements. How many of you are solving questions with me? How many of you are solving questions with me? I do see some very distracted people here trying to build friendships through in YouTube chat. Yes, guys. How many of you are solving questions with me? Yes, please. Yes or no? Ma'am, hide the options. Which song is favorite? Aishu, is this the time to know about her favorite song? Really? Nishant is solving. Never mind. Don't worry about the answers. You'll be getting this PDF. Okay, okay. Now, which of the two statements, competition exclusion principle states that two closely related species competing for the same resource cannot coexist indefinitely and the inferior one will be eventually eliminated. Is this point true or not? Is this statement true? This is absolutely true. This is what Gauss said. Now, in general, carnivores are more adversely affected by competitors than herbivores. No, it is not. It is always the herbivore one which will be more affected because of the limited food options. So, all herbivore can eat is just grass. So, if the grasses are done with, if the grasses are depleted, there will be no other option. Herbivores are very rigid. We will only eat veg. Carnivores are very open minded. So carnivores are less adversely affected by competition. Is this clear? So answer would be A is correct and statement 2 is incorrect. Clear to all? Okay. Next question. Type of interaction. So see the type of interaction. Which one is positive, negative? The first type of question coming from this chapter is type of interactions. Whether which interaction is positive and negative. Then you have also seen questions on birth rate and death rate, right? So these are two most occurring or recurring questions. Now see, mutualism, as I said, is a positive positive interaction. Commensalism is positive zero interaction. Amensalism is negative zero, and parasitism is positive negative. Be it parasitism or even predation, both are of the same type. One is positive, another is negative. Is that clear? Now, logistic growth. Logistic growth is when the sources are limited. So this is limited resources availability condition. Exponential means it will go on just increasing throughout. When can it happen? When the resources are unlimited. Now, expanding age pyramid. So age pyramids are important. Expanded age pyramid is when the percentage of pre-reproductive is the largest, followed by reproductive and the post-reproductive. This is, and stable is when pre-reproductives are equal to the reproductive age group. Are these points clear? Are these points clear to all? Please let me know. Name and date mandatory. I have already helped you for that, Karthik. I made a short. Please go through all the instructions with me. And if you have not filled in your admit card yet, that short is going to help you. That short video is going to help you. So please fill in along with that video. Okay. All right. Moving on. A leopard and a lion in a forest, grassland. So both leopard and lion, what type of interaction will they face? They will be competitors of each other because both are carnivores, top carnivores. So they'll have that competition. Then cuckoo and laying eggs in the crow. So what is cuckoo basically trying to do? That is brood parasitism. And then fungi and roots of the higher plants. They are mutually benefiting each other. And cattle, ergot and cattle, they show commensalism. These are clear-cut examples. So the type of interaction, whether it is positive, negative, positive, positive, minus, minus, or positive, minus zero, as well as the examples of interactions are very, very important. Are very, very important. Now, moving on to the next, A and B. So these are the 2 points they are asking for. Following the growth curve, A represents the exponential growth curve when responses are not limiting, while B represents the logistic growth when resources are limiting. Just did that. Yes or no? So growth models. Yes or no? We've just seen this question. Have we seen this question or not? So 2024 repeated this question. So this is the correct answer. So exponential always gives you J-shaped curve, and logistic will give you S-shaped curve. Yes. Moving on to the next question then. When will the population density increase? So guys, see, it's already been an hour and we are still discussing ecology. Do you see the weightage of ecology which has increased over time? So when will the population density increase under special conditions? So it will increase population density. So net population density will be original population. And what are the factors which are going to increase the population? Births and immigration. And what will reduce it? That is deaths and emigration. Sorry. So this is the formula given. So when the number of deaths exceed the number of births, will it increase the population density when deaths are higher? No. So when number of births plus number of immigrants equal to the number of deaths, and then there will be zero population growth. This is called zero population growth. The number of births plus number of immigrants is more than the number of deaths plus number of immigrants. Okay. When so it says birth plus immigrants and deaths plus immigrants. This is also wrong. What is true? When number of births plus immigrants is more than number of deaths and immigrants, answer would be D. Answer would be D. Yeah. Then guys, we have these different phenomena occurring in nature. Migratory flamingos and resident fishes in South American lakes. What are they doing here? Then abundant tortoise became extinct after introduction of goats in their habitat. What is this? Then Cathameles expands its distribution range in the absence of Balanus. Who can answer this question? 5 closely related species of warblers in different locations of the same tree. So one question, 4 different very important phenomena asked simultaneously. Now see, when the warblers of 5 different species are feeding on the same tree but different locations, so basically this is the resource partitioning. Answer would be B. This is the resource partitioning, yes. And then Cathameles expanding in the absence. This is called competitive release. So when the competition is released, you perform, you outperform. And then abundant tortoise became extinct. This is known as interference competition. So basically, goats were interfering in the grazing of tortoise because goats had higher grazing efficiency as compared to tortoise. And then migratory flamingos and resident fishes, they represent interspecific competition. Then predator, which one of these is a predator? Yes, mutualism, then parasitism and sexual deceit. So very typical example of sexual deceit is shown by a Mediterranean Ophrys, right? It is an orchid, Mediterranean orchid named as Ophrys. Then this is D1. So answer would be here. Mutualism is shown by plant and the pollinator, female wasp and the fig, right? Then we have parasitism is always show, the parasite always shows a disease. So Plasmodium is known for the famous malaria, and then predator is Pisaster. Pisaster is a predator. Now, Connell's elegant field experiment showed that on rocky sea, larger competitors, superior barnacles dominate the intertidal area and exclude the small barnacles from that area. Is that true or false? It is absolutely true. Then generally herbivores and plants appear to be more adversely affected by competition than carnivores. I told you, we have just had a similar statement that during competition, it is always the herbivore which is more adversely affected because of limited food options, right? So herbivores are more adversely, this is also true, but does it explain? No, it doesn't explain. So answer would be B. Is that clear? All right, moving on now. One of this is a certain reason interaction in which one organism benefits, the other is neither harmed nor benefit. So one is benefited, other remains unaffected. What kind of interaction can this be called? This is commensalism. Is this statement true? Yes. Ergots forage close to the cattle. We have already got this example, but in a different format. Yes. So it was in the match the following. We have just solved this in the match the following. So take notice of these repeated examples. Then ergots always forage close to the cattle are grazing. Otherwise, it is difficult for the ergots to find the insects and catch them. This is an example of commensalism. It is not the explanation. It is not the explanation. So answer would be B. Is that clear? Now, population growth growing in an habitat with limited resources will show when it is growing in the limited resources. So what kind of growth model will you get? Logistic. This is a logistic growth model, yes or no? When the resources are limited, we get an S-shaped curve, logistic growth model. Yes or no? Now, lag phase, the phase of acceleration and deceleration, and finally asymptote. So this is how it proceeds. Can you make another question from this one statement? They can simply ask you the correct sequence of phases in the logistic growth model. So it starts with the lag phase, right? Goes on to the exponential phase, also known as phase of acceleration. Then it again slows down and then finally becomes parallel, which is known as asymptote, right? When it becomes equal to the carrying capacity. Now, ability to realize its innate potential to grow in the number and each enormous density in a short time. No, it doesn't. Its ability to realize the innate potential and reach the enormous density in a short time. No, it is not so. And then exponential growth, yes, it will show exponential growth. Logistic growth, I've just said. So answer should be A and D. Sorry, it is not exponential growth. Exponential is one which happens during unlimited resource supply. It's not exponential, it is the logistic. Is that clear? So we get S-shaped curve like this. Answer would be A and C. Clear to all? Clear to all? Yes. Vishwa, that is not exponential, that is logistic. Moving on to the next question. When the fitness of one species is significantly lower in the presence of another species, what do you call this as? What do you call this as? Competition? Yes or no? And when fungi remain in association with the living and plants, they are called saprophytes. Fungi growing with plants is known as mycorrhiza. It is not a saprophyte. Rather, this is a mutualistic response. This is a mutualistic response. How not to panic when seeing plant anatomy questions. How not to panic? I feel confidence, precedes knowledge. This is one thing I've learned by heart. Confidence comes after knowledge. So if you've well learned the chapter, if you are thorough with the concept, then there should be no panic attack on seeing the plant physiology questions, and you still have 2 days, guys. And also, your adrenaline is working to its maximum. Make good use of that adrenaline rush. Make good use of this. Okay, moving on, guys, now to the next question. The equation of Wilhurst-Pearl logistic growth. From this equation, what does K indicate? K is the carrying capacity. K is the carrying capacity. Gauss, see, these exclusion principles are coming back again and again. So Gauss competition Gauss exclusion principle, and also we are getting resource partitioning again and again. Resource partitioning. Yes or no? So, Gauss competitive exclusion principle states that the two closely related species competing for the same resource cannot coexist indefinitely, and inferior one will be eliminated eventually. This is true. In general, carnivores are more adversely affected by competition. This is false. Answer will be true and false. Now, which of the following is the Wilhurst-Pearl logistic growth equation? Yes, which one represents the Wilhurst-Pearl logistic growth? Yes, guys, let me know quickly. So it is dn by dt is equal to. Okay, Vishwa, got it, got it, got it. Now please let me know the answer. So dn by dt is equal to Rn k minus n upon k. Is that clear? Easy to remember. Then this was a controversial question of NEET 2025, right? A controversial question here. A fig fruit is a non-vegetarian fruit. No, it's not. So, as it has enclosed fig wasps in it. But fig wasp and fig tree exhibit mutualistic response as fig wasp completes its life cycle in the fruit and fig fruit gets pollinated by fig wasp. Again, this was a wrong statement. Why? Because it said it is not the fruit that gets pollinated, it is the flower. It should have been flower. So both the statements were considered incorrect. All right. Next, epiphytes, guys, are growing on the mango branch. It is an example of epiphytes are the plants growing on other plants only for the sake of shelter. They will not draw any nourishment. So epiphytes will be benefited. Mango will be unaffected because nutrition is not being withdrawn. Such is the case of commensalism. Now, when gene targeting involves gene amplification, coming on to biotechnology now. So ecology is done. It has taken 1 hour. Why? Because eventually MTA has started loving ecology overall, right? 10 questions from ecology means a lot. Imagine. Now see, next question from biotech. When gene targeting involves gene amplification is attempted in the individual's tissue to treat a disease, what is this known as? So when we are targeting a gene to treat a disease, guys, this is known as gene therapy, biopiracy, safety testing, and molecular diagnosis. It is certainly gene therapy, number one. It is certainly gene therapy. Are these points clear? Now, these options are for the second question. Molecular diagnosis is for diagnosis, as the name says. Safety testing, again, for checking the product before it is delivered in the market. Now, here, talking about, wait, yes, with regard to insulin. Now, insulin is also a recurrent topic coming back again and again, and you will see this in today's video. Now, see, just a second. Okay. All right, guys. Moving on. With regard to insulin, choose the correct options. Okay. C peptide is not present in the mature insulin. Is that true? Yes, it is true. There is absence of C peptide. Insulin produced by our DNA technology has C peptide. No. This is where the real accomplishment of Eli Lilly lies. So they directly produced A and B and then joined them together by creating disulfide bonds. They eliminated this step where they had to produce C first and then remove it. Then the proinsulin has C peptide. This is true. And then A and B are interconnected by disulfide bridges. This is true. So which are the true statements? We have A, C, and D, and answer would be this one. Answer would be this one. Are these points clear? Gene can be used to check the diseases as well to produce medicines. For that, no, ma'am. Yes, Vishwa. Okay, you are right. Statements related to human insulin. See, insulin is another interesting and favorite topic of NTA. So, which statements are correct about genetically engineered insulin? Which statements are correct about genetically engineered insulin? Pro-insulin contains extra stretch of C. Okay, but it is asking about genetically engineered. We'll see to the given options. A peptide B are separately produced in E. coli, extracted and combined by creating disulfide bonds with them. True. Insulin used for treating diabetes was extracted from cattle and pig. Yes. Then proinsulin needs to be processed for converting into mature and functional hormone. Are we talking about genetically engineered insulin here? Some patients develop allergic reactions to the foreign insulin. These are true, but please see, out of all the options, we have to choose which is correct for the genetically engineered, not insulin in general. So answer would be B only. See, only this statement holds true for the genetically engineered. See, proinsulin contains extra stretch, right? But then proinsulin is not produced by genetic engineering. Are you getting that? So, insulin which was extracted from cattle and pig is not genetically engineered. All the statements are true, but you have to find one statement which is for genetically engineered insulin. Are you getting the question? So, this is where the trick is. Most of the students will keep on finding that, okay, I'm getting confused, all the statements are true, but read the question well. It is only asking you for genetically engineered insulin. Is that clear? Then milk of Rosie was nutritionally more balanced for the human babies than natural because it contains the same protein which is human alpha lactalbumin. It is also found in colostrum, which is why we hold colostrum very nutritious. right? Now, in gene therapy, that is ADA deficiency, the patient requires a periodic infusion of genetically engineered lymphocytes. Why? Why does the patient have to go for regular transplant, blood transfusion, so that these genetically engineered lymphocytes can be injected? Why? Because lymphocyte gene isolated from this lymphocytes from patients are grown in culture outside. Genetically engineered lymphocytes are not immortal. This is clear NCERT line. So what does it say? They are not immortal, means they will die after a week or so. So you have to get continuous infusion, periodic infusion of genetically engineered lymphocytes. Answer will be C. Are you getting the statements? Next is gene therapy. What is gene therapy, guys? Gene therapy means correcting a gene which is defected. Correcting a gene which is defected. Then RNA interference is a method of cellular defense. Every eukaryotic cell has this mechanism. So this is cellular defense. Then ELISA is a diagnostic test. What is happening? ELISA is a diagnostic test for AIDS, and gel electrophoresis is a method of separation of DNA fragments. Is that clear? DNA is negative, move towards positive. Are these points clear to all? Please let me know. Quickly let me know. Are these points clear to everyone? Alright guys, biotechnology is the easiest in zoology. That depends, guys. Only if you have learnt it well, you have understood the topic well, you can say so. Which of the following techniques does not serve the purpose of early diagnosis? Early diagnosis for its early treatment. That means which of the following technique is not molecular diagnostic technique? Molecular diagnostic technique, not. So we know ELISA is molecular diagnostic. We know PCR is molecular diagnostic technique. rDNA technology is also molecular. But serum and urine analysis, they are typical or conventional methods of diagnostic for a disease. Is this clear? Now, so these are the conventional methods which select the incorrect statement regarding the chemical structure of insulin. Again, insulin coming back. Mature insulin consists of 3 polypeptide chains. This is itself a wrong statement because mature does not have C. Insulin is synthesized as prohormone with an extra stretch of C peptide. That is true. So in our body, first it is a proinsulin which is produced, which later gets processed to insulin. C peptide is not present in the mature insulin. This is true. And then so correct answer is A, right? Because of the C part, it becomes incorrect. And then polypeptide A and B linked by disulfide bonds, again true, true, true. Is that clear? Let's move on to the next question. Main steps of formation of recombination DNA. Arrange the steps in the correct sequence. You have to find out the sequence. Insertion of recombinant DNA into the host cell. Okay. Cutting of DNA at the specific locations, isolation of desired DNA fragment, amplification. So first of all, you will cut it at the specific location. Then you will isolate that desired fragment. Then you will amplify it. And after making multiple copies, you will insert it in the recombinant DNA. So it has to be it has to be BCDA, BCDA, right? This was a KCET question. All right. Now, moving on to during the purification process of rDNA, addition of chilled ethanol. NTA has just asked this question. Isn't that so? NTA is just asking PYQs and confusing you with few questions as per its nature, right? Chilled ethanol precipitates out what? DNA. Now, gene gun method is used to introduce alien DNA into host. Microparticles of which metal are used? It is tungsten and gold. So how costly is gold and how costly would this gene gun method would be? Now, RNA interference takes place. See, RNA interference method is also another topic coming back again and again. RNA interference. Now see here. RNA interference takes place in all the eukaryotic organisms as a method of cellular defense. This is true. RNAi involves silencing of specific mRNA due to complementary single-stranded RNA that binds and prevents the translation of this. Please let me know if this statement is right or not. Yeah. If you know the answer to this question, I can trust that your preparation has gone really well. Yes, please. I want to see if you can answer this question, guys. If you can answer this question quickly. If you can answer this question, you have prepared well. Let me see who is answering the question here. Nobody yet. Is it so difficult? Really? RNA interference. So guys, it is clearly written in NCERT that specific mRNA due to complementary, it is not single stranded, it is double stranded RNA. So this is where the statement goes wrong. So A is true, B is false. A is true, B is false. Right. प्रोटीन सिंथेसिस को सप्रेस करता है, बिल्कुल करता है, बट यहाँ पे बात बतानी है गलत क्यों है. It prevents translation, that is true, but how? Because it is itself double stranded. Now, moving on to Bt toxin, genetically engineered Bt cotton kills the pest by what is the mechanism in which it is going to act? Creating pores in the midgut, damaging the respiratory system, nervous system, or altering the pH. So, we know that Bt toxin is going to cause perforations in the midgut or epithelial cells of midgut, right? Now, genetic engineered human insulin is produced within E. coli, right? There were two separate E. coli. See, GM cotton is produced with the help of Bt. So basically, GM crops cotton, so that is genetically modified, it contains gene of Bacillus thuringiensis. So A2 and B4, A2, B4, answer would be this. ADA deficiency can be corrected by gene therapy, and ELISA is an antigen antibody reaction. Are these points clear to all? Please let me know. Now, which of the following steps involved in the action of toxin in Bt cotton? Bt cotton. Okay, steps of action. How do you produce that? So, sequence based question again. Inactive toxin is converted into active toxin due to alkaline pH of the gut. Bacillus produces crystals with toxin. Alkaline pH solubilizes the crystals, and activated toxin binds to the surface, create pores, and cause death. And toxin protein exists as inactive prototoxins in bacteria. Now see, it will start with, yes please, let me know which is the correct answer guys here. How is the process going to start? How is the process going to start here? Anyone who can answer, let me see. Quick, quick, quick. Yes please. So it guys starts with Bacillus thuringiensis. Bacillus thuringiensis produces the crystals which are toxic, insecticidal. Then it's going to, this toxin, the toxin which is produced, this toxin is inactive within the bacteria, and that is why the bacteria is not affected by its own toxin, because it is prototoxin, inactive form. Then this alkaline form gets solubilized in the pH medium. This is C. Then it has 4 inactive toxin converted to active form due to the alkaline pH. And finally, the active toxin binds to the surface, create pores, and cause the death of the insect. Is that clear? Is that clear? Wasn't it a smart and a tricky question? Yes or no? Okay. Now, 2024 question. Indian government has set up GEEK, which will make decisions regarding the validity of GM research. Absolutely true. Biopiracy is the use of bioresources by native people. No, if it is used by the own people, native people, then it is not called piracy. We have every right to do that. It is by the foreign people, foreign companies, foreign countries, right? Now, so this is false. Answer is A. Answer is A. Now, Bt toxin, coming back again and again, can kill many insects. That doesn't kill the bacillus. Why? I just told you. Why is bacteria not affected by the own toxin? Because it is present in the inactive state, and only once the insect ingests it, it is converted to the active form, because insect has alkaline pH in the midgut. Alkaline pH. It requires alkaline pH to get active. Now, some ethical standards are required to evaluate the morality of scientific human research that might help or harm. Yes, there has to be some ethical standards. You can't go around making any, discoveries. And then Genetic Engineering Approval Committee, GEAC, makes decisions regarding the validity of research and safety of introducing it for the public services. This is also true. So is the statement correct explanation? Yes, please. Let me know. Is the statement correct explanation? Yes or no? No, it is not a correct explanation. Rather, had the reason been an assertion, and assertion would have been reason, then answer could be A. So the second statement is not able to explain the first statement. Important objective of biotechnology in agriculture: to produce pest resistant varieties, increase nitrogen content, decrease the seed number, or increase the plant weight. So in agriculture, see, we have produced BT cotton, we have transgenic tobacco. So what are these? In both the cases, we are trying to produce pest resistant varieties. Yes or no? Yes or no? So we are trying to basically produce pest resistance. That is the main objective. Now, two DNA sequences are prepared corresponding A and B chains of human DNA were introduced in plasmids of agrobacterium to produce insulin. It was not agrobacterium. The host used for insulin production is E. coli, never agrobacterium. Agrobacterium is preferred in plants. Uh, Plant-based. Now, chain A and chain B are produced separately, extracted, combined by creating. So this is true. So A is false and B is true. Do we agree with all these questions? Are we on the same page, guys? Are we solving these questions? Yes or no? Let me know, guys. Are we solving these questions and able to recall everything? Okay. Now, capacity to generate the whole plant from any cell. This capacity to generate, this is known as totipotency. Now, for example, zygote. Zygote is one totipotent cell, one cell which can give rise to the whole organism is zygote. Now, Bt toxins are insect group specific and coded by the gene Cry1AC. Cry1AC. Now it says Bt toxin exists as inactive toxin B, inactive prototoxin in Bacillus thuringiensis. However, after ingestion, inactive prototoxin gets converted into active due to acidic pH of insect gut. It is not acidic, it is alkaline. And what about the statement number 4? This is true. They are group specific. We have 3 examples, 1AC, 2AB. Is that clear? So they are group specific. Which of the following are fused in somatic hybridization involving two varieties of plants? So in somatic hybridization, guys, there is combination of protoplasts. There is combination of protoplasts. So answer would be C. For example, can you name an example? It is pomato. Pomato. Pomato is produced by potato and tomato by fusion of their protoplasm. You have to mark the incorrect statement. A bioreactor provides optimal growth conditions for achieving the desired product. Most commonly used is stirring type. They are used to produce small scale bacterial cultures and they have agitated oxygen delivery system and foam control system. All the statements are true, but this small word has made the statement incorrect. Bioreactors are the large vessels, guys, 1,000 liters of capacities, right? So it is for large scale commercial production, not for small scale bacterial production. Now again, match the following. So alpha 1 antitrypsin, guys, is a protein produced by rDNA technology, biotechnology, for the treatment of emphysema. For the treatment of emphysema. Are these points clear? Till now, are we clear? Okay. Then it says Cry1AB is going to is going to protect the plant against corn borer, while Cry1AC is going to protect it from cotton bollworm. And enzyme replacement therapy works for ADA deficiency. Answer would be C. Answer would be C. Now, which of the following genetically engineered organisms are used by Eli Lilly to prepare the human insulin? Just told you, Eli Lilly used E. coli for as a host. So what was E. coli? E. coli is a bacteria. It is a bacteria. Clear to you? Okay, silencing of specific mRNA is possible via RNA interference because Why? We've again seen a similar question, but in a different format. So in RNA interference, we make complementary. It is not non-complement. It is complementary, but we make double-stranded RNA. Did I tell you or not? It was a statement-based question in 2024, and then it came out in this format in 2025. We are again getting a question on RNA interference this year. We are 100% getting a question on RNA interference this year. Just the format will be, of course, a different format. We are definitely getting a question on this. Now, next question is specific recognition sequence identified by endonucleases to cut them at the specific locations within the DNA. What is that specific sequence known as? We call it a cloning site, recognition site. What is the specific property? It is palindromic in nature. So it reads the same when read in the forward and the backward direction. Now, during the process of gene amplification using PCR, very high temperature is not maintained in the beginning. Then, which of the following steps in PCR will be affected the first? Very high temperature is not maintained in the beginning, then which of the, okay. It says if, see, we know there are 3 steps. We have, it is not ligation at all. The steps of PCR go in this sequence only: denaturation, annealing, and extension. So denaturation, we know it is going to occur at around 90 to 94 degrees Celsius, right? Annealing at 52 degrees Celsius, 54 or 52 degrees, 54 degrees Celsius, and then extension at 72. So it says if we don't maintain a very high temperature in the beginning, which step will be affected? It is denaturation. Denaturation would not take place. The double-stranded DNA will not break. Is that clear? Now PBR 322 has PST1 restriction site within the gene amplification, within the gene AMPR, ampicillin resistant gene, that confers ampicillin resistance. Okay. If the enzyme is used for inserting a gene of beta galactosidase production and the recombinant plasmid is inserted in E. coli strain, what will happen? It will lead to lysis. It will be able to produce a novel protein with the dual abilities, and it will not be able to confer ampicillin resistance, or the transformed cell will have ability to resist ampicillin as well as produce beta galactosidase. So what will happen? So question is, since, see, let's say this is PBR 322, this is your AMPR resistance gene, and if we use PST1 endonuclease here, and we insert this another gene for beta galactosidase, what will happen? So the ampicillin gene resistance will not be conferred. It will not confer. See that we are doing insertional inactivation. We are inserting another gene. This process is known as insertional inactivation. Yes or no? Insertional inactivation. Are we clear here? Okay, moving on. Which of is the correct sequence? Just discussed. The correct sequence would be first is denaturation, then annealing, then extension. Answer is C. Then, during the purification process of RDNA technology, addition of chilled ethanol precipitates out. NTA has asked this question in 2023, 2021, and has given the exact same question in its WhatsApp group, right? For your so-called preparation or revision. So it has asked this question multiple times. It is DNA. Are we clear here? Are we sorted till now? Please let me know, guys. Are we sorted till now? Okay. Moving on now. The agarose gel electrophoresis. Agarose is a natural polymer obtained from seaweed. Can you name? It is Gelidium G2. That is Gelidium and Gracilaria, the red algae, right? This is true. Then the separation of DNA molecule in agarose is dependent on the size of DNA. Yes or no? Yes. The smaller the DNA, the faster it moves. The DNA migrates from negatively charged to positively charged. Of course, DNA itself is negative, so it will run away from negatively charged electrode towards the positively charged. This is also true. And then it says the DNA migrates from positive to negative. Now this is wrong. So the correct answer would be A, B, and C. Are we clear till now? Are we clear till here? Pathogenic bacteria gains resistance to antibiotic due to changes in. So see, this is a line given in the chapter biological classification as well as in cell. So plasmid confers the antibiotic resistance. What is the significance of plasmids in bacteria? So plasmids have some special genes like antibiotic resistance, fertility, right? Answer would be C. See, it's clearly written. Which of the following method not commonly used for introducing the foreign DNA? It is bacteriophages. Complicated. Is that clear? Answer will be A. Now, gene gun. Again, methods of making or transforming a host. Methods of transformation. Methods of transformation. Now, gene gun. Where do we use this gene gun? Gene gun is used to transfer the gene, and they're preferably used in the plant cells. Gene therapy to replace a faulty gene, yes or no? Then gene cloning, to make cloning means to make the identical copies, and genome is the total DNA in the cells of organism. It is not actually total, it is a haploid set. So answer would be B. Answer would be B. Ma'am, hydrochloric carrageen is obtained from which class? Vishwa, carrageen is red algae. Carrageen. Another is algin. So I have given you a trick also. Carrageen has double R, so it is for red from red algae. Clear? Separation based on both charge and size. No, because all the see, it is not based on charge, Asha, because the whole DNA is negative. How will it get separated then? Right? So it is getting separated on the basis of size. Yes or no? Do you agree? It is only based on size because the whole DNA is negative. So if it is not for size, then the whole DNA is going to move together. Yes or no? Right. Then next question is enzyme A is needed for isolating genetic material from plants and enzyme B for isolating from the fungus. So from plant and fungus. So plants have cellulose and fungus has chitin. So the enzyme used for plant would be cellulase and for the fungus it would be chitinase. It is answer C. Is that clear? This has cellulose, so we'll use cellulase. This has chitin, so we'll use chitinase. Are these points clear? Right. Now, separation of DNA fragments. So, how are we going to separate? It is by agarose gel electrophoresis. Now, we know this. So, the question on gel-based electrophoresis is also coming again and again. So, gel electrophoresis. So, sometimes it is asking the bases and so on. Now, when a particular restriction enzyme cuts the strand of DNA, overhanging structures or sticky ends are formed, right? This is true. Some restriction enzymes cut the strand a little away from the center to the of the palindromic side. This is also true. Yes or no? Yes or no? Please tell me. Are these points clear to everyone? Yes, go ahead, guys. Are these points clear to everyone? Any doubts till now? Quickly. Any doubts till now? Shall we proceed? Okay. Now moving ahead, it says, okay. So these are the two statements. Both the statements are true, but do they explain each other? Let's see. So why do they produce overhanging stretches or sticky ends? Because they cut it slightly away. If they cut right in the middle, guys, if a palindrome is cut right in the center, in the middle, that means 3 on this side and 3 base pairs on the other side, it will not produce sticky ends, it will produce blunt ends. So yes, it explains it very well. Answer will be B. Now, which of the following is not true regarding the gel electrophoresis technique? Not true. So gel electrophoresis is also quite a likable topic by NTA. The process of extraction is known as elution. It says separated or stained by using ethidium bromide. Presence of chromogenic substance gives blue colored bands on the gel and bright orange colored bands can be observed when exposed to the UV light. So answer is what is not true? Answer is C. We don't use blue colored DNA. We see bright orange colored bands and that too when observed in UV light and not under normal white light. Is that clear? Which is not a desirable feature of cloning factor. So yes, features of cloning vectors, another very important uh, topic. Features of cloning vectors. Features of cloning vectors. You should note down. You have to revise this again. Then it says presence of a marker gene. Okay. Presence of a single recognition site. Presence of two or more recognition sites. And then origin of replication. So what are the three conditions we have read? ORI. Any gene attached to ORI will automatically get replicated. Yes. Yes. Then there has to be a selectable marker. So selectable marker is known as marker gene. Selectable marker. So on the basis of which we find out whether the transformation has taken place or not. And third, there has to be single cloning site so that endonuclease cuts it at one place only. So if there will be multiple cloning sites, let's for example say if the plasmid has multiple E. coli 1, sorry, multiple Gattaks, GAATTC, at multiple places this is present. E. coli will cut it at every site wherever Gattak is present. So that's going to cause a lot of confusion. Where are we going to insert the foreign gene if there will be multiple cut sites? So answer has to be this, which is not a desirable gene, sorry, desirable feature. Then restriction endonuclease recognizes specific sequence to cut DNA known as this. True hai? Endonuclease is going to cut at a specific sequence, and that is a palindromic sequence, reads the same in forward and backward direction, cuts DNA slightly away. This is also true. This is also true, but they don't explain each other. They don't explain each other, right? Now, polymerase chain reaction is a DNA amplification. Yes, we make multiple copies. An amplification resistance gene, sorry, ampicillin resistant gene is used as a selectable marker to check the transformation. Yes. So in PBR322, guys, there are two selectable markers. There are two selectable markers, which are TETR gene and AMPR gene. Right? There are two selectable markers here. So both the statements are true, but not correct explanations. So answer will be A. Which of the following acts as a molecular scissor? Such an easy question. It is a restriction enzyme. Now, which of the following is incorrect about Agrobacterium tumefaciens? So, Agrobacterium tumefaciens is used to deliver the gene of interest in both prokaryotic and eukaryotic. No, it is used for delivering the gene of interest in the plant cell. We use this for the plant cell. For animal cell, we are going to use what? Retrovirus. Yes or no? We prefer retrovirus for transferring a gene in the animal cell. Because Ti plasmid is used for gene transfer, not pathogenic to the plant. True. Transfers the normal cells to the tumor cells. Yes. This is tumor inducing. So any plant cell with Ti plasmid will become tumorous. And this plasmid has a special gene which induces tumor, that is tDNA. So the wrong statement is A. Clear to all? Now, ligation of foreign DNA at which the site will result in loss of tetracycline resistance of PBR322. So we have seen, so PBR322 is also another topic which recurs again. Yes or no? Comes back again and again. Okay. Now see, where if you want to insert a foreign gene, in TET-R, what are the two possible locations to cut it? That is by using BAM-H1 and SAL1. So another one is SAL1. These are the two endonucleases we can use to cut the TET-R. Ligation means cutting, ligating it and inserting a foreign gene. And whenever we insert a foreign gene, the property of the original gene is dissolved. So tetracycline resistance will be compromised. Answer will be D. Now, thermostable DNA polymerase used in PCR was isolated from, that is from Thermus aquaticus. That is why this thermostable DNA polymerase gets its name, TAC polymerase, because this polymerase is obtained from Thermus aquaticus. That gives its name, TAC. Main steps in the formation of recombinant DNA are given below. Arrange the steps in the correct sequence. Okay. So first of all, insertion of rDNA into the host cell, cutting of the DNA at the location, isolation of the desired DNA, amplification, and then insert. So it's easy. First of all, we are going to cut it. This is step 1. After cutting, we are going to isolate it. After isolation, we are going to make multiple copies, and then finally we will insert it. This is BCDA. BCDA. Please tell me all these points are clear to you. Please tell me these points are clear to you. Yes, guys. Please tell me all these points are clear to you. Anyone with any doubt till now? Anyone with any doubt till now? Okay. All right. All clear? Perfect, Asha. In gene gun method, I told you, gene gun method is preferred for plant cells. Yes or no? It is preferred for plant cells. And we have already seen this question in the older years, right? So in gene gun, it is you microparticles of which particles are used? Tungsten and gold. Yes. Now during purification process, addition of chilled ethanol. Have we seen this question or not? Have we seen this question or not? What do we purify using chilled ethanol? NTA has asked it this year also. This is DNA. Exposure of UV radiation. See, one is your cloning PBR 322, cloning vectors, and methods of transformation, and then this electrophoresis. These are very, very likable topics. Tetracycline resistance from the previous gene. Okay, I'll explain it. Wait. Let's solve this question and then we'll go by it. So DNA stained with ethidium bromide will show bright orange color, but only in the UV radiation. Ethidium bromide can be seen as bright orange color, but only in the UV radiation. Now, there's a demand of understanding, this topic. So guys, we have PBR 322. So this is how PBR 322 looks like, right? So I'm going to talk about just two selectable markers here. We have two selectable markers. One is ampicillin resistance, and then there is tetracycline resistance, TET-R. Okay. Now, if we want to see any plasmid which will have these two genes will be resistant to these two antibiotics. So they cannot be killed by using these antibiotics. Now, these two are also the selectable markers because we can differentiate between the transformed bacteria and the non-transformed bacteria. Okay. Now, if I want to cut tetracycline and insert a foreign gene, then I can cut it at two places. One is BAM-H1, BAM-H1 as in, see, the way we write BAM-H1 clearly tells you this is a restriction enzyme. This is a molecular scissor. It's an enzyme. And then there is SAL1. So basically these are two scissor molecules, right? So we can cut tetracycline resistance gene by using either of the two scissors, BAM1 and SAL1. And then we have similar two scissors for cutting into ampicillin resistance gene. One is PST1 and PVU1. Are you getting that? If you want to cut it, why do you want to cut it? You want to insert a foreign gene here. Let's say here or here. So wherever you cut, you will be able to insert a foreign gene. Now, if a foreign gene is inserted, what will happen? The tetracycline resistance gene will be manipulated. So the part of DNA which was offering tetracycline resistance is now compromised because you have added a foreign gene into it. Are you getting it? Now this process is called insertional inactivation. You are inactivating the TET-R gene by inserting a foreign DNA. Are you getting that? Please let me know. Anyone? Similarly, I can compromise the ampicillin resistance by inserting another foreign gene by using either PST1 or PVU1. Clear? Yes or no? Okay. Now, moving ahead, which of the following, okay, coming back, all right, which of the following is not a cloning vector? Not a cloning vector. Probe is not a cloning vector. Probe is the one which will diagnose, which will find out what's happening. We use a radioactive probe in autoradiography, while all the other and autoradiography is used to detect a mutated gene. So probe is going to be used here. All right, guys. Moving here to the next question, steps involved in the process of PCR. Again, PCR is another favorite topic. So the steps are in this sequence. Okay. Yes. So first of all, we have denaturation, sorry. Denaturation is followed by annealing, that is addition of primase, RNA primer. Then we will add Taq polymerase and deoxy, and in the presence of Taq polymerase and deoxynucleotides, it will form a long polymer. Extension or elongation will take place, and finally the amplification will occur to 1 billion times. Am I clear here? So answer is C, A, D, E and B. Answer is A. Is this clear? All right. Moving on. Correct about E. coli 1. It cuts the DNA with blunt ends. I've explained you what does a blunt end mean. See, if this is a DNA and let's say there is GAATTC, right? GAATTC. Now we know E. coli is going to cut it here between GA, right? So what will happen? The rest of the portion left will form sticky ends. So whenever it cuts slightly away from the center, a sticky end is produced. This is how E. coli 1 works. This is E. coli 1. But then there are other scissors which cut right in the center. So 3 on this side, 3 on this side. So right in the center when it is cut, then it forms blunt ends. There are no sticky ends. So it's difficult for it to bind. But E. coli cuts forms the sticky ends. That is true. Recognizes a specific palindromic site, and yes, the specificity is it has to read Gattak like this GAATTC. Then cuts the DNA between base G and C. Is that right? It is cutting between G and A on both the sides. Yes or no? And it is an exonuclease. No, it cuts away from the what do you say, the extreme ends. It is in center. So it is not an exonuclease, it is an endonuclease. The correct statements would be B and C and D. B, C and D. Are we clear here? Are we clear here? All right. All right, guys. Moving on. Let's see. Recombinant DNA molecule can be created normally by cutting the vector DNA and source DNA respectively with. Now, the trick is that you must know that both vector DNA and source DNA, both vector DNA and source DNA are cut by the same enzymes. This is clearly written. You don't have to learn uh, palindromic site of Hind or Elu or E. coli or BAM1. So this is just to confuse you. You just have to see that both are similar. Answer is A. So this is where logic comes in. So for that you have to stay very, very calm during the exam and solve the paper. Is that clear? Okay, moving on. Moving on, guys. Oh, wow. There are some students who are actually wasting people's time. The restriction endonuclease enzymes whose restriction sites are present on TETR and in the PBR 322. I've just explained you the structure of TETR. So the enzymes present in the TETR gene, what are those? It is BAMH1 and SAL1. What about these? These are found in AMPR gene. AMPR gene. Then identify the incorrect statement related to electrophoresis. Separated DNA fragments can be directly seen under UV radiation. Yes or no? Directly seen? No. You need to stain it first. So, this is the incorrect statement. Separated can be extracted from gel piece. Yes, this is known as elution. Then DNA moves towards the anode. Anode is positive charge. This is also true. And sieving effect of agarose helps in the separation. This is also true. So all the statements are true, but this is wrong. Hence, this is the answer. Then, if there is a specific DNA sequence responsible for initiating, where is the replication initiated? It is known as ORI. Easy. Restriction endonuclease finds its specific recognition site and binds to the DNA. It cuts each of the two strands of the double helix that is specific in the sugar phosphate bond. Is that true? Is that true? Yes, both the statements are true. It will find the specific recognition site, and it will cut each of the two strands at a specific point. And where are they cut? What does it cut? Sugar phosphate bond. Sugar phosphate bond. That means phospho ester, phosphodiester bond is broken. This is another question, guys. This is another question. What bonds are broken by endonucleases? Which bonds are broken by endonucleases? It is the phosphodiester bonds. Yes, please. Is this point clear? Very good, very good. Now, which of the following? There is one student who's just continuously telling students not to watch these question-based videos. I want to address this. See, I know these questions are already there in your booklets, and I'm sure you have already solved them. Then, what is the significance of every teacher solving PYQs with you on these videos if it is not relevant? Do you think it is irrelevant? The most important questions, guys, for your preparation are PYQs. And please let no one tell you, except your teachers, tell you what to do. No one to tell you. Why are we repeating PYQs at this hour? Because number one, it will help you recollect the whole content. Number two, it will give you an idea what is the most important content to be revised again and again, and also make sure that maybe you have not missed on the important topics. Is that clear? So let no one else tell you all these useless things, and please focus. You don't even have to give clarifications, just focus on your work. Sankalp, ma'am, can I join you too? Where do you want to join me? You already joined the session. Alright, not a selectable marker, not a selectable marker. Ampicillin, metmorphin, chloramphenicol, and tetracycline. Yes, which is not a selectable marker. Which is not a selectable marker. Yes, metmorphin is not, because we use antibiotic resistance genes as cloning vectors here. Sorry, we are using it as a selectable marker here. So it has to be an antibiotic. All these are antibiotics, but not metmorphin. Clear? All right. Moving on. Which of the following is incorrect? Most commonly used is of stirring type. It has been repeated. Used to produce small scale bacterial cultures. Wrong. It is not small. It is large scale production. The same question has come up again. Now, TI plasmid of agrobacterium stands for, TI stands for tumor inducing plasmid. Easy? Then, so this is the question. This is the question. All right, guys. See the question. See this question. Yes, please. So gene X, this is the X gene, is responsible for resistance of the antibiotic, while Y is involved in the replication of plasmid. Then gene X is responsible for controlling the copy number and Y for the replication of plasmid. Then gene X is a protein involved in the replication of plasmid, and Y, yes, and Y is used for resistance to antibiotic. Gene X is for recognition sites and Y is for antibiotic resistance. Please let me know which statement is true. What is the correct role of X and Y? What is the correct role of X and Y? So no, guys, X stands for, let me change the color of the pen. X is the ori. Yes or no? X is the, what is X here? X is the ori, origin of replication. And what is Y here? It is ROP. It is ROP. That is for replication of plasmid. ROP is replication of plasmid. This will help in the replication. If you remember, plasmid also gets replicated along with the nucleoid, right? So this is replication of plasmid. So what is the correct answer? Answer is B. Now, fate of piece of DNA carrying only gene of interest which is transferred into the alien organism. What will happen? The piece of DNA would multiply itself independently in the progeny. Then it says, yeah. Then it says It gets integrated into the genome of the recipient. It can multiply and be inherited along with the host DNA, or alien is not an integral part of chromosome. So it doesn't replicate independently. Alien is not an integral part, it becomes an integral part. It gets integrated into the genome first, then it is multiplied along with the host DNA. So answer would be B and C. Clear to all? Now, hind-2 cuts DNA molecules at particular sites called recognition sites, and they consist usually of, NCERT is written, usually consist of 6BP. Please tell me these points are clear to you all. Why 6? It can be usually 6, but can be 4. So it answer sorry, the recognition site has 4 to 6 base pairs. Yes or no? But usually 6, so we'll go for answer 2. Is that clear? But never more than 6. Never more than 6. Now, the DNA fragments extracted from gel electrophoresis can be used in the construction of rDNA. Yes or no? That is why we are extracting the DNA fragment. Smaller size is observed near anode while larger are towards the wells. So we know that electrophoresis is based on the size of DNA. So the smaller one is the fastest and easily reaches the anode, and larger one will trail behind. So this is true, this is true. Answer would be both the statements are true. Clear to all? Not essential for gene cloning. Do we need restriction enzymes? Yes, we need to cut DNA. Do we need ligases? Yes, they will be useful for binding the DNA. DNA mutase is not needed. DNA recombinase is not needed. DNA polymerase is not needed. So what do we need? We just need A and B. Are these points clear? It is said not essential. Sorry, answer won't be A and B. Not essential is, sorry. We need this. We need this. Yes, please. So answer would be C and D. DNA polymerase is needed. Na? Sorry. DNA polymerase. Endonucleases are DNA polymerases. Endonucleases. Right? So, right. So we don't need these three. For DNA polymerases will be needed for, sorry, PCR. What am I doing? They'll be needed for PCR. They'll be cutting the DNA at specific locations, they'll be binding, and then we'll be using DNA polymerase or tag polymerase for amplifying it. What is not essential? Mutase and this. Answer would be C and D. Are these points clear to all? Please let me know. Are these points clear to all? Yes? Okay. Moving on, guys. Moving on now to the next. Okay. Blue and white selectable markers have been developed which differentiate between the recombinant from the non-recombinant colonies on the ability to produce the color and the presence of a chromogenic substance. The blue colored colonies have DNA insert in the plasmid and they are identified as recombinant. And colony without blue color DNA insert are identified as recombinant. So which is right or wrong? Now we know there is this enzyme called beta-galactosidase. Remember? This enzyme is going to produce blue color. A bacteria has this. If a bacteria has this enzyme, it will produce blue color. So basically, without blue color is identified. If it is not able to produce blue color, it means it has been manipulated. It has been engineered. So without blue colored, it will be recombinant. This is true. And the blue colors are not recombinant. They are non-recombinant. They are the originals. So this is wrong and this is true. Answer would be this. Is that clear? Okay. All right, guys. Moving on to this question, this is the bioreactor identifier which is used as a foam breaker. So first of all, you need to know what the labeling here. Let's label. So it says foam breaker. Foam breaker is C. This is the foam breaker. This one is the foam breaker. Can you label rest of these? Let me know in the comments or in the chat. Yes. Next questions are polymerase amplifies the DNA. What is the formula? What is the formula to see how much it will be amplified? Answer would be 2 raised to power the n. N is the number of cycles. Number of cycles. Yes or no? Okay. Now, right, Jia. B is motor. Right. Moving on then. Now, this is another form, see 2025 question, another form in which they have given your a plasmid. In a represented plasmid, an alien piece of DNA is inserted at E core I one site. So this is E core I from here to here, it says, and we are inserting another piece of DNA. And what does that do? Which of the following strategies will be chosen to select the recombinant colonies? And see, in this E core I1, the enzyme that we are going to put is beta galactosidase. The gene that will be inserted here is beta galactosidase. So how are we going to choose the recombinant colonies? Yes, please. How are we going to choose the recombinant colonies? Yes, please. Very important question and very interesting question. See, beta galactosidase is going to produce blue color. Yes or no? It is going to produce the blue color. Right? But the ones which have been, so beta galactosidase produces blue color. Now, if an alien piece of DNA is inserted, what will happen? What will happen? This blue color will not be produced. So white colonies will be produced here and they will be selected. So the ones which are white are the recombinant. So recombinants will not produce this white color there. Are these points clear to all? Guys, moving on to the next topic, which is microbes. Very important chapter. You usually get 1 to 2 questions every year. Aspergillus niger, citric acid. Yes, acetobacter, as the name says, acetic acid. Butylicum rhymes very well with butyric, and lactobacillus, we all know lactic acid. Let's speed up for this chapter now. Microorganism identification responsible for the production of immunosuppressive cyclosporin A. So cyclosporin A is produced by Trichoderma polysporum. Yes. So I always remember it as Miss TC. So Monascus will produce statins and Trichoderma will produce cyclosporin. All right. Antibiotics are chemicals produced by microbes that kill other microbes. They are chemicals produced in the body that eliminate the microbes. Antibodies are chemicals produced. So this is true. Antibodies are produced in our body, but antibiotics are chemicals produced by microorganisms to kill the organism. So both are correct. Both are correct. Now another match the following. Streptococcus is going to produce what? A clot buster known as streptokinase. TC, I just told you, MSTC, Miss TC. So Monascus purpureus is going to produce statins. Trichoderma is going to produce cyclosporin A, and Aspergillus niger is a citric acid producer. Then aphids and mosquitoes are natural pests. Is that true? Yes. Ladybird and dragonflies are natural pest controllers. This is also true. In fact, ladybird will control aphids and dragonfly will control the mosquitoes. Are these points clear to all? So both the statements are true. Now, Saccharomyces is known for the production of ethanol. It is a brewer's yeast, right? Then Aspergillus niger, coming back again and again, citric acid. Trichoderma polysporum, as I told you, is cyclosporin A. Clostridium butylicum is going to produce butyric acid. Clear? Now, Bacillus thuringiensis, bacterial infection by Bt can kill only caterpillar larva. Now, Bt cannot kill the adult moths. Are these points true? Yes or no? This is true. This is true. They are not meant to kill the adult moth, but the caterpillar stage. Clostridium butylicum, once again, the name goes with butyric acid, Saccharomyces, ethanol, Trichoderma, Miss TC is cyclosporin, and this is streptokinase. Please, are you able to solve the questions before me? Are you able to solve these questions? Are you faster than me? You have to be, guys. Right? Okay. Which are not involved in the preparation of household products. So this produces citric acid. Citric acid is not a household product. Bacillus produces curd. It is a household product. Trichoderma polysporum, just told you, cyclosporin. It is not a household product. It is a bioactive molecule. Saccharomyces produces ethanol, or it helps in the formation of bread. So bread we can consider is a household product. Propionibacterium helps in the formation of cheese, Swiss cheese. So it is a household product. It says not involved in the production of household. So answer should be A and C. Is that clear? Should be A and C. Clear? All right. Moving on. Streptokinase. Bioactive molecules are very, very important. That whole paragraph. Chemicals, enzymes, and bioactive molecules. Bacteria streptococcus used for, it is a clot burster, guys. Fibrolytic activity. Remember? For myocardial infarctions. If moth and caterpillar are of same species, no, still not. These biocontrol agents, they're very specific. Not only are they specific on the target insect, but also the specific stage which they are going to destroy. Clear, Shwin? All right. Which of the following is a non-distilled alcoholic beverage? Non-distilled is beer. The other example would be wine, while all the others are distilled forms and will have higher alcohol content. Moving on now, ADA deficiency results into ADA deficiency is going to cause SCID. So lack of immune system, neither AMIS nor CMIS. So dysfunction of immune system. Is that clear? Now, why? Because ADA is required for the maturation of lymphocytes. So neither B or T will mature. Now, coming on to health and diseases, what does filariasis? So, which organism causes filariasis? So, it is caused by the filarial worm, Wuchereria. Amoebiasis by entamoeba. Pneumonia, also caused by Haemophilus or Streptococcus pneumoniae. There are two names of the pathogens given. Ringworm is a fungal disease caused by Microsporum, Trichophyton, and Epidermophyton. So, they can give the exact same matching, just giving a different microbe here. Yes or no? Identify the incorrect pair. Ricin is a drug. Codeine is an alkaloid. Yes or no? Toxin. Heparin is a toxin. And lectins are concavalin. Please tell me that which of the following is a wrong statement? Which of the following is a wrong statement? Yes. Yes, guys. Which of the following is a wrong statement? This should be easy. This should be easy, guys. Anyone? So which is the correct answer? That means the incorrect pair is ricin, drug. Ricin is not a drug, right? Okay. Effective treatment of the disease, early diagnosis, and understanding of the pathophysiology is very important. Which of the following is a molecular diagnostic technique used for the detection? So we know there are three molecular diagnostic techniques we have studied. One is ELISA, second is PCR, and the third one that we have read is autoradiography. And also the Western blotting, right? ELISA is followed by Western blotting. Now, which of the following is not, is very useful in early detection? Okay, which of the following is very useful in early detection? It is ELISA technique. Is that clear? Okay. Now, barriers. They are the non-specific. So cellular barriers are cellular barriers are your WBCs. Cellular barriers are your WBCs, which are phagocytic, neutrophils, monocytes. Cytokinins are interferons which work against virus. Physical barriers is a mucus, cilia, hair, and skin. They are physical barriers. And what about physiological? HCl, earwax, tears, saliva, right? Then lactic acid in vagina. Are we revising all these examples along with the questions? Yes or no? Are we able to revise? What about Western blotting? It is, Veshvi, it is a technique, but the most popular, the question said most popular. See here, is very useful for early detection. So the Western blotting is followed. It is done after ELISA. Clear? So ELISA is more popular. What's Western blotting and Southern blotting? See, Southern was the name of the scientist basically. Rest just followed the rest. For rest of the names, we just followed the directions. So Southern blotting is for DNA. Southern for DNA. Northern for RNA. Western for proteins. And Eastern for modified proteins. So these are different techniques. Okay. Okay. So Southern was the name of the scientist. Rest we just followed the Wait. Okay. Moving on. Which of the following reasons is mainly responsible for graft rejection? It is cell-mediated immune system. And can you tell exactly which cell of T cell-mediated? It is T killer or T cytotoxic, which is responsible for graft rejection. Moving on. Incorrect statement, guys. Incorrect statement with respect to acquired immunity. Anamnestic response is elicited on subsequent encounters with the same pathogen. Anamnestic response is secondary immune response, which is true. This response will be generated. Elicited means generated when we encounter the same pathogen for the second time, third time, and so on. Anamnestic is due to the memory of the first encounter. This is also true. This is very, very true. Acquired is non-specific. That is the incorrect. Acquired is a very specific response. Now, primary response is when body encounters a pathogen for the first time. Now, can anyone tell me which type of antibody is produced during the anamnestic response and what about the primary response? Let me see. Let me see. How many of you know the answer? How many of you know the answer? Yes. Which antibody is produced during the secondary and primary responses? Yes, please. I'll move on to the next question. Autoimmune disorder is a condition where body defense recognizes its own cells as a foreign bodies. True. And then rheumatoid is the condition where body does not attack the cell. It does attack. It does attack your synovial membrane. Synovial membrane, which produces synovial fluid, that gets attacked. So first is true and second is incorrect. Is that clear? Okay. Moving on now. Again, a repetition of a similar question. See, cytokinin barriers. Right, Jaya? So primary has, no, primary immune, let's go back to this question, guys. Primary produces IgM and in secondary it is IgG. It is IgG. D is never produced in such responses. Now, cytokinin barriers are the interferons. Cellulars are the, I told you, phagocytic cells, and these two are neutrophils, monocytes, macrophages, all phagocytic. Physiological, tears, saliva, earwax, HCl, lactic acid. Physical barriers, mucus, cilia, hair, and skin. Now, moving on to the next question, diacetylmorphine drugs is known as, diacetylmorphine is known as heroin, also known as smack. It is an opiate. It is an opiate. Where is it developed from? It is developed from morphine. As the name says, diacetylmorphine. So all you have to do is diacetylation of morphine will give you heroin. Now, typhoid is caused by Salmonella typhi. Elephantiasis, again, by Wuchereria. Now, Wuchereria is a Eschelminthes. Ringworm is because of a fungi, Microsporum. In the previous year, it has given another fungal organism, fungus, sorry, and malaria is due to a protozoan. Can you do all these questions? Are you able to recollect all these questions? Ringworm, again, Trichophyton. Filariasis, again, Wuchereria. Malaria, due to Plasmodium vivax. Pneumonia, due to Haemophilus. What is another organism which causes pneumonia? Give me the name of the organism which causes pneumonia. Guess, guys. Quickly, give me the name of another organism which can cause pneumonia. Yes, please. And guys, I hope you know that there are still 2 more days left for AIMT before the D-Day. Go for these mock tests. Let's not have any regrets later. Regrets don't work. Hemophilus influenza to diya hua hai. It's already given, Kingi. It's not influenza. That is already there. My question is, which is another organism responsible for causing pneumonia? It is Streptococcus pneumoniae, right? Vaishnavi, very good. Now, talking about the drugs, heroin. Heroin is an opiate, hence it is a painkiller. Marijuana is a cannabinoid, and cannabinoids have effect on vascular system. Cocaine is a, cocaine is that. It is a stimulant and it interferes with the transport of dopamine. It interferes with the dopamine receptors. And then morphine. Morphine is again an opiate and depressant and it will slow down the body function. But no. So we have made it wrong. Okay, sorry. See, heroin is not going to be used. Heroin will slow down the function and morphine will be used as a painkiller. Sorry. Marijuana will if have effect on the cardiovascular, and cocaine will interfere with the dopamine receptors. Can we do that? A2B1, A2B1, C4, and this is the answer. Clear? Yes. Now moving on. Commonly sexually transmitted disease is completely curable, which is completely curable STD. Completely curable STD. It is gonorrhea. See, I've already given you an answer. How to remember the incurable diseases. Incurable STDs. All that you have to learn is 3H. What are the 3H? Herpes, hepatitis, and HIV. So which one can be easily treated if detected early? The condition is it has to be detected early. Yes. Now, which of the blood cells HIV undergoes in replication and produces? So basically, which cells are used by the HIV as factory cells? As factory cells. Yes, guys. Actually, it is macrophage, but the second one is B lymphocytes. Since we don't have an option of macrophage here, we'll go by B lymphocytes because after macrophages, they're going to attack B. Now, B lymphocytes, guys. Yes, please. B lymphocytes. What do B lymphocytes do? They will produce an army of proteins in response to pathogen. And what do we call that army of proteins as? Antibody. Yes or no? We call it antibody. What's happening here? It is called antibody. Okay. Yeah. Antibodies. Then interferons. Interferons provide you innate antibody, innate immunity. That is cytokinin barrier. T lymphocytes, part of cell mediated. And then colostrum, you get directly made, ready-made antibodies. So it provides you passive immunity. So D1, A3, answer will be C. Are these points clear to all? Now moving on to the tumor, malignant tumor, guys. What is the characteristic of malignant? What do we mean by malignancy? Cancer. What is the characteristic feature of this malignant tumor which differentiates it from the benign tumor? It is that it spreads, and this is known as metastasis. What is a MALT? It is a secondary lymphoid organ, not primary, it is secondary, and more than 50% forms most of the 50% of secondary. NACO is a National AIDS Control Organization. It's an NGO which works on various aspects related to AIDS. Alpha interferons, they are anti-tumorous. Are these points clear? Isn't it the helper cell? So, yeah, that's what I said, no? Sorry. Yes, you're right. Guys, we have made this mistake here. HIV, the factory cell used by HIV is a macrophage. When it is not given, then it has to be T helper cell. Sorry, it is not B lymphocyte, it is T helper cell. Absolutely right. Thank you. Yes, you're very right, Jaya. You're right. Now, moving on, because T helper cells are the managers. Once you control T helper, everything is under control. Very right, right. Okay. What type of question is this? Ma'am, can a question come that when was first bio NCERT published? Why would they ask this question? When they have better questions to ask and they know that you can be easily tricked into questions from NCERT. Why do they need to give you any such question? This question in Pancham Maheshwari intro that first NCERT published. Oh, no. If you want to learn, you can, but there are better questions. Yes, they can definitely ask such questions. They can, why not? Okay, coming on to this question now. Some human organs like the, got it, SD, got it. Learn it. Some human organs like liver and kidney fail to function normally, and transplantation is the only remedy to enable the patient to live a normal life, right? Then tissue matching and blood group matching are essential before undertaking any transplant. The patient has to take immunosuppressants thereafter. Are these true? Both statements are absolutely true. But do they explain? Some human organs fail to function normally, and transplantation is the only remedy. This is right. Because tissue matching, no, because it doesn't make sense. Answer will be Both true but not correct explanation. Is this clear? Is it clear? Okay, what do I think about the level of NEET 2026? I think we should be more concerned about our level. That's that's the best I can say. We should be more concerned about our level, guys. Are we well prepared? So NTA has given a slightly difficult paper in 2025, or you can consider in the last 5 years, 2025 paper was difficult, right? So it has already shown its colors. Plus, NTA is marked by its unique property of giving surprises. So we don't know how it's going to happen. Maybe it's very easy, maybe it's very difficult. So we'll wait and watch. The point is, our level should be right. We should be well prepared, not wasting time. And my level was bio was done, but I was so anxious. I didn't get you. All right. All right, guys. Typhoid. Typhoid is caused by Salmonella typhi, which is a bacteria. Leishmaniasis is caused by Leishmania. It is a flagellated protozoan, right? Ringworm is by a fungus and filariasis is by a roundworm. They've asked about filariasis multiple times. Bone marrow is a main lymphoid organ where all blood cells, including lymphocytes, are produced. Absolutely true. Now, both bone marrow and thymus provide microenvironments for the development and maturation of T lymphocytes. In the light of above, choose the correct answer. Give me an answer to this question. Yes, guys, give me an answer to this question. Yes, please. Quickly, give me an answer to this question, guys. Okay, watching you. Give me an answer to this question. Both the statements are right? Absolutely right. They give microenvironment where they develop. Right. Now, which of them are autoimmune diseases? Myasthenia gravis, yes, it affects it affects your acetylcholine receptors. Then rheumatoid arthritis, your synovial membrane. Gout is a metabolic disease. Muscular dystrophy is a genetic disease. And then systemic lupus erythematosus. Yes, lupus is an autoimmune disease. So answer would be A, B, and E. Are these clear? Okay, all these examples are given in NCERT. Cocaine. Cocaine is produced from Erythroxylum coca. Heroin is an effective, uh, no, heroin comes from poppy, which is Papaver somniferum. Morphine, again, it has asked a similar question in 23, is an effective sedative. Marijuana, obtained from cannabis. Clear? Common cold, also known as rhinitis, will be occurring because of rhinovirus. Hemozoin is involved during malaria, comes from the burst out RBCs. So, Plasmodium Vidal test is a diagnostic test for typhoid and allergy due to dust mites. Then, not correct. Antigen binding site is located on the C terminal. So, guys, this is your antibody molecule. And on this side, we have N terminal. This is the C terminal because we have COOH groups here. So, C terminal. So, it says antigen binding site. So, where is antigen going to bind? It is going to bind on the N terminal. So, this is the incorrect statement. Constant heavy, region of heavy and light chains are located at C terminal. So of the antibody molecule, which is right. So variable is towards the N and rest of them are C. So they are towards the C terminal. Each has two light, two heavy chains, true. And then they are connected through disulfide bonds, all true. Then coming on to type of immunity present at the time of birth and of non-specific type. Okay. So we will call that as innate immunity. We have different names: innate, inborn. We also call it congenital because all are synonyms. Congenital. We also call it genetic because genetic is something you're born with, right? Genetic. Coming on to the next. Lymphocytes migrate for interaction with the antigens to the secondary lymphoid organ tissues. Where do they migrate? For interaction. So they migrate to the secondary organs, secondary tissues, right? So which of these are secondary? This is primary, this is primary, these three are secondary. Answer would be C, D, E. Clear? Then computed tomography, CT, and MRI scan for detecting cancer of internal organs. Yes, true. Therapeutic drugs, chemotherapeutic drugs are used to kill non-cancer cells. Why would we take that? Alpha interferons They help in they activate the cancer patient's immune system and destroying the tumor. Yes, they work from inside. Drugs are biological response modifiers. In case of leukemia, cell counts are decreased. No, they are increased tremendously. So answer would be A and C. A and C. Is this clear? All right, moving on. Neoplastic. Okay, neoplastic characteristics of cells are. What is neoplasty? It is another term for tumor. Okay, neoplastic characteristics of cells. A mass of proliferating cell, rapid growth of cells, invasion and damage to the tissue, and confined to the original location. So basically, neoplastic is the benign one, right? So it proliferates, rapid growth, but invasion. Invasion is a character of cancerous or malignant. So it remains, so answer would be A. Then adaptive radiation. Let's move on to evolution, guys. Adaptive radiation example is Darwin's finches. Convergent evolution is seen by analogous organs, wings of butterfly. These are analogous. Analogous, yes or no? Analogous shows convergent. Divergent is shown by homologous organs. These are the examples. And anthropogenic action, evolution occurring to anthropogenic action, is resistant varieties developing because of excessive use of herbicides and pesticides, even antibiotic resistance. Are you able to answer these questions? Please let me know that you are able to recollect. Please let me know you are able to recollect. Yes, guys. When will you say second year chapters? Second year chapters? Which second year chapters? Jyoti? Okay. Then founder's effect. The factor that affects the founder's effect is genetic drift. So we know, see, Hardy-Weinberg equilibrium is disturbed by, it is disturbed by all these factors, right? Now, founder's effect is basically when a part of population separates from, drifts away from the main population. So when a part of population is drifting away, it is getting reproductively separated. What will happen? It will carry the alleles along. So the allelic frequency is going to change. So change in the allelic frequency is called genetic change in allelic frequency. That will be the founder's effect. Okay, now which is not true? Analogous results in convergent evolution just seen. Potato, sweet potato are analogous. Homology indicates common ancestry. Flippers of penguin and dolphins are a pair of this homologous. No, this is a bird, this is a mammal. They are not homologous. So all are true, but not this. Moving on, natural selection where more individuals acquire specific character value other than the mean character value. So means not the intermediate. So organisms, nature is selecting the individuals which acquire a specific character other than mean, not the average, but any specific. Yes, guys, let me know the answer to this question. This can be very interesting. What is the answer to this question? Quick, quick, quick. Yes, please. What is the answer to this question, guys? It is directional. It acquires specific character value. Now, other than mean can be directional or disruptive. Now, why the answer is not disruptive? Why? Because it says specific character. It can be one. If it says more natural selection where more individuals acquire the extreme characters, then it means both the extremes, right? But here a specific can be only one, so it is moving on either of the two sides, that is direction. Clear? Correct statement regarding the mutation theory. Large differences due to mutations arise gradually. No, mutation believes in sudden spontaneous changes. It can't be gradual. Theory was given by Alfred Wallace. No, it was Hugo de Vries. Variations are small directional changes. No, these variations are large, non-directional, spontaneous, random. We call them non-directional. So answer is single step, large mutation is a cause for speciation, and this mutation is known as saltation. Yes or no? Do we remember this term? Saltations lead to speciation. Now identify the fossil of a man and the following character. So brain capacity is 1400 cc, so which can be Neanderthal. And Neanderthal was which we started using hides or the animal skin. So from the cranial capacity itself, see, cranial capacity is a unique property of every ancestor. So if you remember, you can easily find. And then it is confirmed by these other features. They bury their dead bodies, use hides. So hence answer is Neanderthal. Neanderthal was the first modern man. Homo sapiens neanderthalensis, right? Then, correct group of Australian marsupials exhibiting adaptive radiation. So you have to learn all the 10 names given in that. So numbat, spotted cuscus, and flying phalange. Also, you have to learn the names given in the table of convergency. Now, just understand this. These names, this table given of convergent, given on convergent evolution. So one side we have placental mammals, another side we have Australian marsupials. So all the organisms in one line, they show adaptive radiation amongst themselves. Similarly, all those in this will also show adaptive radiation amongst themselves. But if we compare these two, that is when they show convergent evolution. Yes or no? Or adaptive convergence. Are you getting the point? Answer would be C. Now, with respect to convergent evolution, let's see. So this question is from that table. So you have to find the correct pair. So lemur goes with spotted cuscus. Bobcat goes with Tasmanian tiger cat. Anteater goes with numbat. And flying squirrel goes with flying phalange. So you have to learn them in pairs. Learn in pairs. Only if you have learned it in pairs, you can answer this question. Now, this famous figure of an evolutionary phenomena, what phenomena does it show? It is adaptive radiation of Darwin's finches. So, you have done two examples of adaptive radiation, one of Darwin finches and second of Australian marsupials. Yes or no? Gene pool. What is the gene pool? Total number of genes and the alleles. Drift, change in the gene frequency. Just told you. What is genetic flow? Transfer of genes into and out of the population. When we move, we carry our genes along. Gene frequency is stable within a generation. Now that is called Hardy-Weinberg equilibrium. Are these points clear? All right. Moving on now. Coelacanth caught in the South America is an example of vertebrate transition from. Yes. So what type of transition does it represent? Yes, please. What type of transition does it represent, guys? So it is a transition of fish to amphibian. Do you remember? Came out on land and can go back to water. So what else it can be? It can be fish to amphibian. Yes or no? Now, which of the following will not affect the Hardy-Weinberg equilibrium? Genetic recombination, genetic drift, gene migration, and constant gene pool. If it is constant, then it is an equilibrium. These all will affect. They will disturb. There are 5 factors given which will disturb Hardy-Weinberg equilibrium. Are these points clear? Now, moving on to the next. Wait. Yes, guys. Let's move on to the next. So we have these eras, geological time scale given here. Geological time scale given here. Let's see. So in which event occurred during which era? So answer is Mesozoic era gave us birds and reptiles. Proterozoic era gave us lower invertebrates. Cenozoic era gave us mammals and Paleozoic era gave us fish and amphibia. Now there can be another type of question. Arrange the time scale in the increasing order or from primitive to current. So do we know that this is the current era going on? This is the most recent era. We are living in Cenozoic era. Most recent. Yes or no? So the first one is Proterozoic. Then we have these fish ones, Paleozoic. Then we had Mesozoic, and then we have Cenozoic. This is the most recent. Are these points clear to all? Now moving on to stages of human evolution arranged in correct sequence. Yes. Listen. So answer would be, first of all we had Homo habilis, then Erectus, then Neanderthalensis, and then Sapiens. Clear? Answer would be D. So human evolution is a good topic. Now coming on to flippers of penguin and dolphins. What type of evolution do they show? Told you these are birds, these are mammals, so they are analogous. They are analogous to each other. Yes or no? And what type of evolution is shown by analogous organs? Convergent evolution. Now, sweet potato and potato, again they are analogous. Why? Because same function but different structure. This is a stem, this is a root, but function has to be same. So the function is food storage. Yes or no? Correct combination would be, it is analogous, and analogous always show convergent evolution. Clear? Now AUG codes for methionine and phenylalanine. Please tell me, are these points clear? So far, are the points clear to all? Quickly let me know. So far, the points are clear to everyone. Alright guys. Now moving on to AUG. AUG, it says codes for methionine and phenylalanine. Yes or no? Does it code for methionine also? Sorry, code for phenylalanine also? Yes? Yes. It generally codes for methionine, but it codes for phenylalanine also in some prokaryotes. Now AAA, AAG are both going to code for lysine. Both going to code for lysine. Sorry. Doesn't code for phenylalanine. Now we are just going to talk about the universal. So AUG only codes for methionine, right? But this is AAA, AAG. They code for lysine. Yes, they code. So answer would be B. Answer would be B. Which of the following statements about histones is wrong? So histones are rich in amino acids like lysine and arginine. Yes, they are. They are basic amino acids. They carry positive charge. Yes, basic does carry positive charge on ionization. Histones are organized in an octamers, that is unit of 8 molecules. This is also true. They are, the pH is slightly acidic. No, the pH is basic. They are alkaline because of the lysine and arginine. The only enzyme that has the capability of catalyzing initiation, elongation, as well as termination during the process of transcription. Very good. What is transcription, guys? DNA to mRNA or RNA. That is transcription. All types of RNA. That is transcription. So DNA dependent RNA polymerase. Yes or no? So from DNA to DNA, we'll have, this is used during replication. This is used during replication. So from DNA to RNA, so DNA dependent RNA polymerase, it is going to catalyze all the 3 steps, starting with initiation, then followed by elongation and termination. Just the difference is that during initiation, it will require a start factor, which is sigma factor. And during termination, it will require a stop factor, which is a rho factor. So sigma plus polymerase initiation and sigma plus rho termination. For elongation, it doesn't need any other factor. Which of the RNA is not required for protein synthesis? Not required. So we need rRNA because they are going to form ribosomes. We need a messenger molecule. We need a tRNA molecule, right? We don't need siRNA. We don't need siRNA. Please tell me that these points are clear. So what is siRNA? So they'll be more involved during gene silencing. They'll be involved in gene silencing, RNA interference. Now adenine makes 30%, A is 30. See here. And what is the percentage of rest of them? So according to Chargaff's rule, A is equal to T. So this is also 30. So A and T together make 60. The remaining is 40, which will be distributed amongst 20 and 20, guanine and cytosine. So what will be the answer? Answer will be D. Are these points clear? Now identify the correct statement. Coding strand. It says coding strand in transcription unit is copied to mRNA. The coding strand? No. So the one which is copied into mRNA is known as non-coding. We call it non-coding or the template strand. Is it clear? The template one is called non-coding. Split gene arrangement is a characteristic of prokaryotes. Split gene means there are introns plus exons. So that is split gene. That is not a characteristic of prokaryotes. It's a characteristic of eukaryotes. In capping, methyl guanosine triphosphate is added to the 3' end. Let me know. Is it added to the 3' end? No, capping occurs at the 5' end. Tailing occurs. See, it is easy to remember. T for tail, T for 3, right? So tailing occurs at the 3' end. Can you remember it like this? Can you remember it like this? So capping will occur at 5' end, so wrong. RNA polymerase binds with the rho factor, just told you, to terminate the process. Now this happens only in prokaryotes. Answer will be D. Clear? Now, role of RNA polymerase 3. It's very easy to remember the role of RNA. Which are the 3 things which will be transcribed by this polymerase? So, it is 3. It is going to produce 3 things. So T for 3, T for tRNA. And then there are 3 things which are transcribed. Another is 5S, and then it is sRNA. It's easy. So, it has to be tRNA, 5S, and snRNA. Clear to all? So I'm giving you the tricks along. Complete the flow chart of central dogma. So we know what is A? It is replication. B, transcription. C, translation. And what is formed in the end? Proteins. Answer will be A. Now again, Chargaff's rules. So A is 30 and C is 20. What is the percentage of T and G? So we know A and T are equal. G and C are equal. So T would be, it has asked T first. T will be 30 and G will be 20. So this is in accordance to Chargaff's rules. Yes or no, Chargaff's rules. Now, against the codon 5' to 3' UAC, what will be the sequence of NT codon? So on mRNA, we have the sequence 5 to 3' end. Yes or no? So when you have UAC like this, this is the mRNA coded. What will be the anticodon for UAC? So again, it follows the rule of complementarity. So just go with the rule of complementarity. See, rule of complementarity will apply during replication, during transcription, not during translation, but when tRNA binds to mRNA, then rule of complementarity will work. Clear? But not during the genetic code. Genetic code does not go by a rule of complementarity. Please see that. Is that clear? So against U, what will it be? A. This will be U. This will be G. The answer will be AUG. Is that clear to all? Is that clear to all? Please let me know. Okay. Now this is another data that you must be thorough with. So see, this is a question from 2022. So be thorough with this. Bacteriophage 5174, guys. 5174 has these many nucleotides. Lambda is going to have 48502. E. coli will have 4.6. And human haploid content will have 3.3. So it is saying haploid content. What about the 2n content? Just multiply 6.6 into 10 raised to power 9. Is that clear? So just watch the word haploid or diploid. In lac operon, very important. In lac operon, guys, what does I gene codes for? I gene codes for repressor. Then we have Z, which codes for beta. Yes, it codes for beta. Then Y for permease and A for translocase. DNA polymerase catalyzes the polymerization only in one direction, that is 5 to 3. Is that true? Is that true? Yes, it polymerizes in 5 to dash only in one direction. Yes, polymerases. See, it is saying polymerase will only bind to 3' end. It binds to 3' end. I'm not saying polymerization. It binds or reads. It reads in 3' to 5' direction. But polymerizes in 5' to 3' direction. Every polymerase, be it DNA polymerase or RNA polymerase, they both have similar feature. What do we mean by read? We mean that it is going to bind with the OH of 3' end. I hope you know 3' end has alcoholic group. So polymerase will bind to that. It always reads in 3' end, 3 to 5', and polymerization only occurs in 5 to 3. Is that clear? Alright. During replication, one strand, that replication is continuous and other is discontinuous. So this makes both the statements true. Clear? Alright. For lac, just had this question. Z will code for beta galactosidase. Now, 10 E. coli cells with N15 double-stranded DNA, now see very carefully, are incubated in the medium containing 14S nucleotide. All right. So how many? 10. After 60 minutes, how many E. coli cells will have DNA totally free from 15? Totally free from 15. How many will have totally free from 15? Yes, please. Let's see. How do you solve it? So guys, the trick is, first of all, you must know that the replication time for E. coli is 20 minutes. Yes. So it is going to divide in every 20 minutes. How many times minutes have they given? 60. So that means if it divides every 20 minutes, by 60 minutes it has divided how many times? 3 times. Yes or no? So number of cycles are 3. Yes or no? 3 times it has divided. Please tell me. Is that clear to all? So if we are starting with 10 E. coli, then how many E. coli will be produced after three divisions? So 10 will give 20, 20 will give 40, and 40 will duplicate to form 80. Is that clear? So answer would be 80 E. coli will be produced. Yes or no? You don't have to consider 15S or 14S here. Sorry, in the first step. You don't have to consider. First of all, you just see how many E. coli will be produced in 3 cycles. So number of cycles is total duration upon 20. Clear? So after 3 cycles, guys, we'll have 80 E. coli. For now, just consider the time. Now we will see what to do with 15 and 14N. Is that clear? Now see, what will we do? See, we know that 14N nucleotides will appear after 2 generations for each lineage. So it means we will be left with 60 cells which will contain 60 E nuclei which will contain only 15. Why 15? Because see 50. So let me describe like this. This is 59, this is 59. How many parental strands do we have? Only 2. So how many will have the E. coli will have DNA totally free from 15? So it will be 60 cells. Is that clear? Okay. Moving on now. E. coli strand I gene gets mutated. Now in 2022, this question was also much discussed. Now see very carefully. I gene gets mutated. Guys, this is also a very good question of NEET 2022. I gene gets mutated. What was I gene producing? Repressor. What is I gene responsible for? Repressor. So its product cannot bind with the inducer. So we know I gene is producing repressor, the process. But when it is mutated, now it is a mutated repressor. That is why it cannot bind. It cannot bind with the inducer molecule. What is the inducer? It is lactose. Now, if the growth medium is provided with lactose, what will be the outcome? What will be the outcome? Yes, very interesting question. So we know when I gene is producing repressor and when lactose is present, it binds with it. Now repressor cannot bind with operator. This cannot happen. So operator is free. When operator is free, it will stimulate the promoter gene, and when promoter is free, RNA polymerase can bind with the promoter, and this will activate ZYA genes. So this happens when lactose binds with repressor. But now it is saying repressor cannot bind with the repressor. What will happen? If lactose will not bind with the repressor, then repressor can go and bind with operator gene. Yes or no? Yes or no? So when repressor can again go back and bind with operator gene, then RNA polymerase will not bind to promoter and the ZYA will not be able to translate. Are these points clear? Please tell me that this question is clear to you all. Please tell me that this question is clear to you all. Is that clear? Is that clear? Okay. Now, if geneticists use the blind approach for sequencing the whole genome, see there are 2 approaches where we are only going to sequence the transcribing region and then we can sequence the whole region. If we sequence the whole region, it is known as sequence annotation. And if it is not the blind approach, we are only sequencing. See, for whole organism, it is a sequence annotation. But if we sequence only the exons, that is the part which can express, that is known as expressed sequence tags. Can we get this question in 2026? Can we get this question in 2026? Yes. Now, if the total length of DNA is 1.1 meter, what is the approximate base number of base pairs? So guys, we know that when it is 2n, the length is around 2.2 meter. So if this is 1.1 meter, what is the number? So we can go by this also. Yes or no? Okay. Next, DNA polymorphism forms the basis for. Yes, please. It forms the basis for. So DNA polymorphism forms the basis for DNA fingerprinting as well as genetic mapping. Read the following statement and find the correct statements. Find the correct statement. Answer is A. Okay. Euchromatin is loosely packed. No. Sorry. Yes. Euchromatin is loosely packed. That is why it is transcribable. Heterochromatin is transcriptionally active. No. This one is active. The one which is loose can transcribe and active. This is tightly packed and that is why cannot be transcripted. Histone octamer is wrapped with negatively charged DNA nucleosome. Yes, DNA is negatively charged. This is true. This is true. Histones are rich in lysine arginine. This is also true. Typical nucleosome has 400. No, it has how many? How many? 200 base pairs and not 400. Are these points clear? So this is also wrong. Answer would be A. Clear? The process of translation of mRNA to proteins begins as soon. So it is a small subunit which encounters the mRNA first. So first small subunit will bind and then the larger subunit will bind it gradually. Next question is salient features of genetic code, guys. Salient features of genetic code. Is it palindromic? No. UGA is initiator? No. It is AUG. It is unambiguous and specific. Is that true? Yes, it is true. So every code is going to bind for only one type of amino acid only. This is specificity means degeneracy. Yes, not degeneracy. This is called non-ambiguity only. Specific and unambiguous are one and the same things. And then code is nearly universal. Yes, it is nearly universal. Answer would be D. Then guys, moving on to Hershey and Chase experiment. What does Hershey and Chase experiment say? Viruses were grown in the presence of radioactive phosphorus containing radioactive DNA? Yes or no? So if we added radioactive phosphorus, what will become radioactive? DNA or proteins? Phosphorus is present in DNA, so this is true. Grow on radioactive sulfur to form radioactive proteins? Yes. Radioactive sulfur will get added into proteins. Radioactive phosphorus proteins? No. So radioactive sulfur in DNA? No. Radioactive protein contained radioactive DNA? No. So if there is protein radioactive, then DNA also? No. Answer would be this. So it is P32 and S35. Is that clear? Okay. Moving on. Inducer of lac operon. Which is the inducer? It is lactose. That is why it is an inducible operon. RNA being unstable mutates at a faster rate. This is true. RNA can directly code for the synthesis of proteins, hence can express itself. Hence it can express itself. Yes, quickly. Yes or no? Please let me know. Is this true? Yes, this is true. See, instead of forming DNA to RNA and then proteins, RNA can directly code. So both the statements are true. Are these points? Sorry, both the statements are true, not this. Answer is C. Clear to all? Alright. Now see again. P32S35. Just told you which scientist conducted this method. Which scientist conducted this experiment? Yes. Who were responsible? It was Hershey and Chase. When they unequivocally announced that DNA is the genetic material, it was this experiment. Process of copying genetic material from one strand of DNA into RNA. This is transcription. Is that true? Yes. Transcription unit has three regions: promoter, structural gene, and terminator. Absolutely true. So answer would be D. Moving ahead, component that binds to the operator gene of the operon and prevents the RNA polymerase from transcribing. Just explained to you what binds. The component that binds to the operator is the repressor. Last chromosome in Human Genome Project which was sequenced was chromosome 1. It was the largest chromosome. That is why it is rank 1. And there were so many genes that it took a longest time for it to be sequenced. RNA mutates faster. True. Viruses have RNA genome and shorter lifespan, mutate faster and evolve faster. This is also true. So viruses that contain RNA, retroviruses, HIV, COVID, they have a shorter lifespan and they evolve faster. Both the statements are true. Please tell me that these points are clear to you all. Please tell me all these points are clear to you all. They're not a cloning vector. Which is not a cloning vector? PBR 322, probe, back, or yak. Probe is not a cloning vector. Probe helps in determining what is happening. Like it helps in finding out the mutated gene. Rest all are the cloning vectors. Then, how many different proteins does the ribosome consist of? Yes. How many different proteins does the ribosome consist of? So, guys, there are in all 80 distinct proteins. Is it clear? How many? 80 distinct proteins. Moving on now. The corresponding coding strand is given here. Coding is the one against which mRNA is not produced. See that it is a coding strand. If the sequence of mRNAs formed is as follows, which of the following is a sequence? So mRNA is given here and you have to find the coding strand. You must know, guys, mRNA is coded against the non-coding strand. So non-coding and mRNA, they are complementary. But mRNA and coding, they are similar. They are same. They are not complementary. So what do you have to do? Nothing. Just see the same one. So it is, and the only difference would be that uracil will be replaced by thymine. That's it. Otherwise, it will read the same. So A. U. Instead of U, you will get T. Rest C. It will read the same. This is the coding strand. So remember, so when you have to find the coding strand, you have to find out the same sequence except U should be replaced with T. And if you have to find the non-coding strand from the given mRNA, you follow the rule of complementarity. Are these points clear? Now in prokaryotes, the positively charged DNA is held with the negatively charged proteins called in the region called nucleoid. Is this statement true? No. This is negative and this is positive. So this is a wrong. In eukaryotes, negatively charged is wrapped around the positively charged histone octamer to form. This is true. So answer is false and true. Answer is T. And then, so again, you're getting Y. A is going to go for transacetylase. Y is going to go for this. P is not the I is for repressor. Sorry. Here we have I for repressor, Z for this, Y for permease, and A for transacetylase. Are these points clear? Answer will be B. EST, just told you. One, we just did this question. See here. Where was this question? We did this question here. I think it's left far behind. Okay, never mind. Let's move on. Okay, what is expressed sequence tag? One is sequence annotation, in which we sequence the whole genome. But expressed sequence tag, we only code the genes that are expressed as RNA. All genes? No. All genes expressed as RNA. So yes. as RNA? No. All genes that are expressed as proteins. Why? Because only the exons are going to code for proteins. Please tell me that these points are clear to you all. Please tell me these points are clear to you all. Yes, please. Is that clear? Sorry. We're doing it wrong. We're doing it wrong. EST, yeah, expressed annotation. So we are going to code those genes which are going to be expressed as functional RNA. Yes. So it is exons here, not proteins. Yeah. Answer will be A. Clear. Is that clear to all? Is that clear to all? If we include exons plus introns, then it is sequence annotation. All sequences. But only exons, then it is this. Now, moving on to the role of, we have again got this question on RNA polymerase 3, and I told you 3 for tRNA, and then 5S, and then this. Answer will be B. Moving on to unequivocal proof. Just told you who gave the unequivocal, Hershey and Chase. Histones are positively charged because they're basic. Nucleosome, DNA wrapped with histone. Euchromatin, loosely bound. So it is active in transcription, and heterochromatin, densely packed. In eukaryotes, guys, there are 3 RNA polymerases in the nucleus, right? RNA polymerase 1, 2, 3. So eukaryotes have different type of polymerases in addition to RNA polymerase found in the organelle. And all the 3 in the eukaryote have different roles. This is true. Both are true. Both are true. Clear? Now, in lac operon, guys, another favorite topic, Z codes for beta, ZYA. This is a sequence, right? Z codes for beta galactosidase, which is primarily responsible for hydrolysis of lactose into galactose and glucose. This is true. Galactosidase, it is going to break it down. Yes or no? Yes or no, guys? Are these points clear? Right? So this is true. In addition to lactose, glucose and galactose can also induce the lac operon. No, this is wrong. It is only lactose that can induce. Answer will be A. Moving on, in prokaryotes, RNA polymerase is capable of catalyzing the process of elongation. Can it do that in prokaryotes? Can it do that in prokaryotes? Yes or no? It can? Yes. In polymerase, it associates transiently with rho factor to initiate. No, it is not for initiation. It is for stopping, for termination. It is a stop factor. So, A true, B false. Please tell me that these points are clear to you all. Please tell me all these points are clear to you all. Right? Shall we move on? Okay, guys. Moving on, let's stop it here after that. I gene codes for repressor. Now, Taylor and colleagues proved that DNA replicates semi-conservatively. And what did they use? Faba beans, Vishya faba. Vishya faba. It is a eukaryote. Vishya faba. Now, which of the following techniques was used to elucidate the double helix model? So they used X-ray diffraction method. Right? Technique, they are asking for technique. It is X-ray diffraction. Not a characteristic of genetic code. Triplet, yes. Nearly universal, yes. Has punctuations? No. It is comma-less. Some amino acids are coded by more than one codon. Hence, this is called degeneracy. True. DNA fingerprinting, Alec Jeffrey, pneumococcus, then E. coli and bacteriophage. Who used bacteriophage? This E. coli was used by Maselson and Stahel. Yes. Give me the correct answer. Yes, guys. Give me the correct answer here. So, this is Alec Jeffrey. Then we have pneumococcus. Pneumococcus will go with Griffith. Streptococcus pneumonia or pneumococcus. They are known for causing pneumonia. That is another. Then E. coli. E. coli will go with Maselson and Stahel. And then bacteriophage is used by Hershey and Chase. Is that clear? Lactose present in the growth medium is transported to the cell by the action of, yes, who takes it into the cell? It is permease, as the name says. It gives the permission to enter. Just remember. Are these points clear? Are these points clear to all? Please let me know, guys. Now see here, Tata box is not really in syllabus, but these are in, these Tata boxes uh, are involved. They are important promoters. Splicing is done by SNRPs during RNAi. For termination, we need a rho factor and 3. RNA polymerase 3 has been asked multiple times. Now see, correct product of DNA dependent RNA polymerase to the given template. So you are given the DNA template. This is the DNA template. You have to find out the product. What is the product of DNA dependent? So mRNA will be produced. Yes, product of this RNA polymerase is formation of RNA. Yes, please. So first of all, and the only thing you have to remember is you have to follow the rule of complementarity. Simple as that. You have to follow the rule of complementarity. DNA is in 3 to 5 direction. It will read in 3 to 5 direction and polymerize in 5 to 3 direction. This is the answer. They are complementary. Which of is correct regarding the process of replication in E. coli? What is correct? During replication, we are going to use DNA dependent RNA polymerase, DNA polymerase, and polymerization occurs in 5 to 3 direction. Just remember, be it DNA polymerase or RNA, it will read in 3 to 5 and polymerize in 5 to 3. Second last question, guys. Griffith experiment will help in studying the transformation. Jacob and Monod, they helped in studying the lac operon. Hargobind Khurana deciphered the genetic code along with who? And then Michelson and Stahl studied the semi-conservative nature. The transcription unit in DNA has 3 regions. Respect to upstream to downstream. Now upstream means 5' downstream is 3' Let me know the answer, guys. Now, this is the last question of the day, and you are going to answer me in the comment section. If you have read well, you can answer. So, upstream, I'm giving you a hint, is a 5' and downstream is a 3' Transcription unit in DNA has 3 regions. So, what would that be? What would that be? So, first promoter, structural, and then terminator, or it would be the opposite. I'm already giving you a hint, and let me know in the comment section. All the very best, guys. I'll be giving you some short videos also to revise your syllabus. Make sure that you do that. Okay, we'll continue. Thank you so much and keep revising.